describe organic reactions and enzymatic reactions, which is actually also an organic reaction, but also show how synthetic molecules can be employed in biological studies. And without synthesis, we would not gain the knowledge that we have gained. So let me first introduce the players of today, that are the mono and oligosaccharides. Let me take the laser pointer. So here you see glucose. Glucose is a C6 sugar, 1, 2, 3, 4, 5, 6. It's the most abundant organic molecule on Earth, and all the hydroxyls are in an equatorial configuration. There are many isomers of glucose. So here you see galactose, the C2. The C4 is axial, and mannose, the C2, is axial. There are also sugars that lack a hydroxyl. So here you see L-fucose. And today we are going to focus a lot on N-acetyl-neurominic acid, or sialic acid. It's an unusual sugar with 9 carbons. It has a carboxylic acid and an acetamido derivative. These monosaccharides can be assembled into oligosaccharides. They cover the surface of every cell and have been implicated in many, many biological processes. So today I will focus on how cell surface oligosaccharides or glycans are involved in viral infection. So basically, most respiratory viruses and also viruses that infect the gut have carbohydrate binding properties. And that should not come as a surprise, because the epithelia, where viruses infect the host, is covered by a thick carbohydrate layer. And so these viruses first need to attach to mucus, and mucus is basically a very highly glycosylated protein. Then it needs to penetrate this mucus to find a structure on the cell surface that it has to employ for binding and entry. So it's important to realize that glycans, even of closely related species, differ a lot. And this is important, because in that way, species protect themselves from cross-species infection. So when an animal virus becomes a human virus, zoonosis, those viruses need to adapt to the glycome, the carbohydrates of the new host, humans. So we have an interest in understanding this viral receptor specificity evolution and try to exploit that knowledge for surveillance, prevention, and intervention strategies. Studying these carbohydrates is actually quite difficult. If you think about it, if you go from DNA, RNA to proteins, this is all template-mediated biosynthesis. And we are very good at expressing proteins, make mutations, knock out genes. That works all very well. Complex, I should add one more thing. Because these are big structures, we often depict them as a symbol. So a circle is a hexose. Blue has the stereochemistry of glucose. The sialic acid is often depicted as a purple triangle. You can depict these chemical structures also in symbols to make it a little bit more readable. So the point that I was making is it is actually very difficult. There are hundreds of structures on a cell surface to investigate one at a time how they may interact with the virus. So the approach that we take is a bottom-up approach, a synthetic approach. Can we first of all analyze the glycans on a given cell of interest? Can we make a representative set of molecules? Can we place these on little glass slides, covalently attached, to develop a so-called glycan microarray? And can we use this to probe binding specificities of either viruses or proteins? So in this way, we can learn which virus binds to what carbohydrate. This is all biology on a glass slide. The next step that we are taking is, can we actually place synthetic glycans on the cell surface and look for gain of function. We can genetically remove a whole class of glycans, bring them back one at a time, and learn which one is really important for infection. Well, this sounds easy, but it's actually quite difficult. These glycans are very complex. There are hundreds of structures on a cell. And not so long ago, just to make one N-linked glycan could easily be a PhD project. So what we have done over the years is recognize that all these carbohydrates have certain core structures that are then modified by different epitopes at the termini to create synthetic diversity. So what we have done is develop methodologies where we synthesize the core and then through chemical or enzymatic means diversify, get collections of glycans. And we have now gone so far that we can actually automate this. We use a lot of enzymes. And the hallmark of automation is synthesizing on a resin. That does not work well for enzymes. So what we have done is we have molecules that we are going to extend by enzymes. We have a tag that allows the enzymatic modification to do in solution. Then we catch it on a resin, and we do it in such a way that everything can be washed away except the molecule, release it in such a way that it can be used for the next enzymatic step, and do this over and over again. And we have shown that through robotics, we can make very complex compounds. And our efforts have resulted in a large collection of molecules, well-defined molecules, ranging from N to O glycans, polylactosamines, heparan sulfates. But today I'm going to focus on O-acetylated sialic acids. These are the sialic acids that I just mentioned a minute ago, C9 sugars. And I'm going to, and they are modified by acetyl esters. And it is a class of molecules that have been very difficult to study. And I'm going to discuss the importance of these molecules in the context of infections by coronaviruses. Everyone is familiar with SARS-CoV-2, a coronavirus. It has a spike, but it's actually important to realize that 7 coronaviruses have entered the human population, SARS-CoV-2 being the most recent one. So 20 years ago, SARS-CoV-1 infected the human population, but there have been viruses like these OC43 and HKU1 that entered the human population more than 100 years ago. So this happens over and over again, that coronaviruses jump from an animal species to a human. So these viruses use proteins on the cell surface, like ACE2, to bind and enter the cell. But every coronavirus has also a domain that binds to a carbohydrate. And the model of infection is very much that the carbohydrate is employed for initial attachment and to migrate through this thick carbohydrate layer. And when it reaches the cell surface, it finds its protein receptor, and that is then used as a hook for entry. So today I will focus on these 2 viruses, OC43 and HKU1. These are beta coronaviruses. And quite surprisingly, here we only know a carbohydrate that is used for binding and entry. But I will show at the end of my talk that that was actually a wrong notion. And they bind to these acetylated sialic acids. So here you see that in a little bit more detail. So here you have the virus. And the virus expresses 2 proteins, a spike, and this spike binds to acetylated sialic acids. So here you see the chemical structure. There is an acetyl, in the case of humans, on the C9 position. The spike binds and use this for attachment and cell entry. So a virus attaches, enters the cell, it replicates. At one point, it needs to leave. And if it then sees its receptors again, it might get stuck. So the way this virus got around it, it also expresses an esterase. So this esterase can cleave this acetyl ester. Now we have free sialic acid that doesn't bind to the spike anymore, and the virus can escape. This esterase is an interesting protein. Yeah? So it has a catalytic domain that cleaves acetyl esters. It has also a lectin domain, and it actually binds to sialic acids. So this is important. So the enzyme can hook on to where its substrates are and then begin cleaving away these acetyl esters. And we have shown that increases the catalytic efficiency a lot. So let's look at these acetylated sialic acids. So here you see N-acetylneuraminic acid. And this N-acetylneuraminic acid can be linked to a larger glycan in different ways. To the 3' position of galactose, to the 6' position of galactose or galactosamine, but also it can be linked to itself, 2,8 linked to another sialic acid. So the first level of structural diversity is differences in linkage. And then these molecules can be further derivatized by O-acetylation. So we can have an acetyl on C4, on C7, and C9, and combinations thereof. For example, 7,9. And this type of modification is very species specific. We make a lot of 9-acetyl. Bovine makes a lot of, cattle, a lot of 7,9-acetyl. And mice make a lot of 4-acetyl, with other acetyl esters also being possible. So it's believed that we all changed our sialic acids to be a barrier for cross-species infection. So the question that we asked was, so you have many beta coronaviruses. OC43, HEKU1 are the human viruses, but there are canine, bovine, many other viruses. And we wanted to explore how they bind to sialic acids and whether differences in acetylation and linkage type is actually a barrier to species infection. OC43, this human virus, is derived from an animal virus, cattle, and we wanted to learn how did it change binding to these sugars to learn the trick to infect us. That sounds easy, but it's hard. So we need to synthesize these sialosides, not only differently linked, but also having different patterns of acetyl esters. These acetyl esters are quite chemically labile, but they can also migrate easily to a nearby hydroxyl, making them very difficult to handle. And basically, before we started this project, these molecules were called Cinderella molecules. We knew they were there, but very difficult to study. So I was lucky having a very talented PhD student, Zeshi Lee, and he came up with a chemical strategy to make this sialic acid linked to the 3' position of galactose, the 6' position of galactose, or the 8th position of another sialic acid. And what he did is he made a triacetylated species. So we have an acetyl ester on C4, C7, and C9. And what he actually did is he exploited the esterases of these viruses to diversify these molecules. So the bovine coronavirus expresses a heme agglutinase esterase that cleaves very selectively the 9-acetyl ester. So then, if we use this enzyme, we get a molecule that has acetyl esters only on C4 and C7. Under mild conditions, we can make this migrate and do it again. Okay? There is also an esterase from mice that cleaves the 4-acetyl. And in combination with the use of this enzyme, you can make quite a few derivatives. So here is Jack Zeshi Lee, and through chemical synthesis, he made this molecule, which was quite challenging. He installed an anomeric linker so that eventually we can attach them to surfaces. And so he called these molecules quite efficiently. And now, using recombinant enzymes, we could make from one compound many different derivatives. So as I already said, this bovine coronavirus, hemagglutinin esterase, cleaves this 9-acetyl to give this derivative. Under mild basic conditions, the C7-acetyl esters jumps quantitatively to the 9 position. We get this derivative. And now we exploit again this bovine hemagglutinin esterase to cleave the 9-acetyl. And we have this compound. If we use another enzyme, that of mice, we can cleave the 4-acetyl ester, giving us a 7-9 derivative. The bovine enzyme takes off the 9-acetyl. We get a 7 that we can make migrate to 9, and we get this compound. So in one project, just one PhD student, we were able to chemoenzymatically synthesize any possible sialocyte, having any possible acetyl ester pattern. All the molecules have biotin at the anomeric center, and the biotin allows us to immobilize these molecules on the surface for binding studies. So that's what we did. We generated a microarray. So we have a glass slide that is covered by streptavidin. And then through piezoelectric printing, we can, on one little glass slide, print all our synthetic molecules. And then we can come in with a protein or a virus of interest. We let it bind, we wash. And then with the detecting antibody with the fluorophore, we can look which of the oligosaccharides lit up and are actually the binding patterns. So here you have this human coronavirus, OC43, and its ancestor is a bovine virus. So here you have the microarray data of the bovine virus, of the spike, as well as the hemagglutinin esterase. And the way it's organized is blue are all the molecules that are alpha-2,3 linked, yellow are alpha-2,6 linked, and green alpha-2,8 linked. And here we have the different patterns of acetyl ester modification. So you can quite nicely see, if we look to the spike of bovine, it's quite promiscuous. When it jumped to humans, it becomes very, very selective. It only prefers this disilaric acid and a little bit of the alpha-3. So the spike has changed quite a bit of specificity when it became a human virus. Also, something happened to the hemagglutinin esterase. So the bovine virus can bind to carbohydrates. Those of humans cannot, because there is now a carbohydrate very close to the catalytic domain that basically blocks it off, and the lectin domain doesn't work anymore. And the result is that this enzyme is much less catalytically active. And the biological reason is probably that we have much less acetylated sialic acids. So if this enzyme works too hard, the receptors are gone before the virus can infect the host. Okay, so we look to a lot of viruses. So here you see 3 human viruses, OC43, HKU1, and influenza C. They all bind to acetylated sialic acid. And you can see they have all one molecule in common. That is a disialoside modified by a 9-acetyl ester. So we think all these viruses have evolved to this structure. And we have evidence that this is quite prevalent in the upper airways as a receptor for binding and entry. If you look to viruses that infect other species, the pattern is very different. And probably that has evolved to recognize molecules where the virus infects these animals. So we looked at lots of coronaviruses that infect different species. And we looked at many different glycans that we synthesized. And you can see that each species has a unique panel of binding specificities, indicating that all these viruses have adapted their binding specificities to that of the host. And humans, human viruses, are quite unique in recognizing this structure, this disaccharide with a 9-O-acetyl ester. So where do we find these molecules? We actually find them on gangliosides. These are glycolipids. There we have two sialic acids linked to each other. And there is one enzyme that is critical for the biosynthesis of this compound, and that is ST8-SIA1. Okay? So we did biological experiments. We took HAK cells, these are kidney cells, and we look at infectivity of OC43 and HKU1. So these viruses are able to infect kidney cells, but if we overexpress ST8-SIA1, we see an enormous increase in infectivity. HKU1 cannot infect these cells, but if we overexpress this critical enzyme, we see a lot of infection. This enzyme is critical in making the acetylated sialic acids. If we knock that out, we lose all infectivity. So biologically, we confirm what we found, that these are critical receptors for these viruses to bind and enter cell. Okay, so we are now able to synthesize these 2,8-linked sialocytes quite nicely, and we wanted to understand at the molecular level how they interact with the spike. And so we were already able to recombinantly express this spike molecule. We can synthesize these dicellulosides. So with cryo-EM, in collaboration with my colleague Daniel Hurdes, also at Utrecht University, we looked at binding. And here we got lucky. So we have the spike, and when we add this sugar, something remarkable happens. So now it binds. And now this, after sugar binding, the spike opens up. And now we see 3 new domains. And this was actually quite revealing, because as I mentioned in my introduction, all coronaviruses, they use a sugar for attachment, and they use another domain actually to bind to a protein for cell entry. And when we saw this spike protein opening up, we thought that actually this may be the domain that binds to a cell surface protein for entry. So this structure is just to tell the virus, you've entered the cell surface, open up, find your protein target, and enter the cell. So computationally, we investigated how this dicelluloside may actually trigger opening of the spike. And actually what we discovered, when this sugar binds, there is a shift in a peptide sequence that basically is like a spring, and the molecule opens up. And we did that in collaboration with Martin Frank in Sweden. Well, quite remarkably, in the same issue of Nature, another laboratory actually identified this protein receptor that is important for entry. That is this protein TMPRSS2. And this is actually the same protein that is being used by SARS, by MERS for cell entry. So it looks like a small number of proteins is being used over and over again for cell entry. Okay, so we can make these molecules now quite nicely, but it is still difficult synthesis. And so we ask ourselves, these CMP sialic acids with acetyl esters, can we synthesize these more easily? And then again, we look to how does nature do it? And so the way nature does it, it has sialic acid, it puts on CMP, and then there is a protein called Cas-D1 that puts an acetyl ester on. And then this is employed by cellular transferases to modify all kinds of oligosaccharides. This protein Cas-D1 has 7 transmembrane domains. We could not express it. So then we ask, can we synthesize these CMP sialic acid with acetyl esters? And that is something that we accomplished quite recently. We synthesized these thioglycosides. We used a trick to diversify these compounds with hemagglutinin esterases and migration. And then we can hydrolyze this thioglycoside, make the sugar nucleotide. And now we can make a whole panel of these molecules chemoenzymatically. And now we can use them not only to make relatively simple oligosaccharides, but we can also employ these donors, for example, CMP sialic acid with a 7,9-O-acetyl ester, to prepare highly complex N and O glycans and even glycolipids. So that's the end of my story. I hope that I convinced you that with chemoenzymatic methodologies, we can make highly complex glycans that are further modified by acetyl esters. And now that we have these panels of oligosaccharides, we can learn host specificities of viruses. We can learn how a virus has learned the trick to infect humans. And we hope to use that as predictions, how future viruses may learn to bind to certain sugars that are important for host infectivity. So these are the people who contributed to the study. With Raoul de Groot, he's an expert in these beta coronaviruses, and he gave us all the recombinant spikes. And Daniel Hurdes, with him, we did the cryo-EM studies, and these are people in my lab who performed the various experiments that I described today. And thank you for your kind attention.
Yeah, the audience can applaud, Geert-Jan, but they will not hear that. Thanks very much for a highly interesting lecture, beautiful work. And I see questions coming up in the Q&A box, so I will now take a look. Or I see people raising hands. Oh, yeah, you get also applause on your screen. Maybe you.
Oh, thank you. I see it. Thank
you.
You
can now see it.
A virtual applause.
So for the attendants, if you have a question or a comment, please put it in the Q&A box, and then I will read it out for you. I have, of course, the privilege then to start with a question, Geert-Jan, how I can imagine that it's very difficult if you study these enzymatic deacetylation to actually determine what product you have, because all these molecules are really very closely related. Is this an NMR study, or do you do this by mass spectrometry?
Both. So by NMR, we can very easily, you need high field NMR and some expertise, but by NMR, we can basically assign every proton, and acetyl esters are strongly electron withdrawing, so you get upfield shifts. So by NMR, we really can identify these molecules. But we have also mass spec-based approaches in which we combine ion mobility with mass spectrometry. And all these molecules have different drift times in IMS, and that gives us an additional means of characterization. But NMR is the go-to technology.
Yeah. So these reactions are also done on an NMR scale, so to speak. So milligram scale.
Yes. A milligram is the smallest amount that we would synthesize.
Oh, okay.
They are all being prepared in multi-milligram quantities.
Okay, thank you. And there's a question by an attendee, which is a bit related, because the attendee asks how to avoid acetyl migration during synthesis.
Yeah, very good point. These are tricky molecules, and what we learned is that under mild acidic conditions, pH 6, these acetyl esters are perfectly stable. If you go to neutral conditions, especially the acetyl ester on C7 has a propensity to migrate. So it depends a little bit on the acetyl ester. The 4 is relatively stable, the 9 as well. The 7 is very tricky, but if we keep it mildly acidic, they stay nicely in place.
Thank you. More questions or comments? I see that's not the case, so then I thank Geert-Jan once again for his beautiful lecture. And then we move on to the next speaker, which is Professor Jana Reuthova from Radboud University Nijmegen, and she will tell us more about mapping reaction intermediates in electrocatalysis. Jana, floor is yours.
Thank you very much. I have to find
Yeah, I see already a presentation coming up.
Yes.
Though not yet in full screen. Yeah, now I see it in full screen mode.
Okay, so thank you very much. It's a great pleasure to present here and highlight part of mechanistic research that is done in the Netherlands. So I will focus today on mapping reaction intermediates in electrocatalysis, but I will start with a little bit more general introduction. So this is these are people in my group, and they are all working on developing some new methods for investigation of reactions, and we mostly focus on developing methods in mass spectrometry. So in connection with investigation of reaction mechanisms in organic chemistry, you might ask, why should we use mass spectrometry? I mean, we heard that when you do the research of sugars, NMR is the tool to go. And mass spectrometry admittedly has some disadvantages. So it is ex situ method. So you have to transfer your species, intermediates, reactants, products from the solution to the gas phase where you detect it. And you detect only ions, and not all of the species you are interested in are ions. However, I will try to show you that there are huge advantages that may prevail over these disadvantages. So firstly, mass spectrometry is extremely sensitive and allows you to detect intermediates that you cannot detect with other methods. There is enormous development, so you can use different methods to transfer your species from solution or from the surface or from the medium where you do the reaction to the mass spectrometer. And there's also enormous development in analyzers. So we have a better sensitivity and better resolution with which we can have certainty in elemental composition of the ions. There are new dimensions that are coupled with mass spectrometry. So you heard in previous lecture already that mass spectrometry can be connected with ion mobility, and then you can connect elemental composition also with basically shape of the molecules. But I will today show you that we can also connect it with optical spectroscopy, which allows you for each ion to get infrared or UV spectrum, and allows you to really assign the structure and electronic structure. And then there is also a development how to bridge the gap between the solution where most of the reactions happen and the gas phase detection. I will not talk about this, but these are also methods that we develop in my group, and they allow you, for example, for organic reactions, organometallic reactions, to estimate the lifetime of the reactive species in solution while detecting them by mass spectrometry. Okay, so I'll go to the first step in investigation of reaction intermediates, which is detection of reaction mechanisms, which is detection of reaction intermediates. You can do it in a very simple way, just connecting your reaction vessel, reaction flask, under the conditions under which you perform the reactions. So it can be stirred, it can be heated, it can be cooled. You just connect it by a capillary directly to a mass spectrometer and continuously flow a small amount of the reaction mixture to a mass spectrometer. This is suitable for most of the organic reactions and most of organometallic reactions that have good kinetics that allow you to detect intermediates in this way. If you go to inorganic chemistry, where we usually work with more reactive metal complexes, you have to shorten the reaction time to detect reactive intermediates, and there we work with flow reactors. You can shorten the reaction time to seconds and study reaction intermediates by changing the conditions in the flow reactor. And this can be automated. You can, in programmed way, study concentration gradients, and it allows you to have a look at kinetics connected with the intermediates that you detect. You can also study reaction intermediates in photochemistry, and today I will focus on reaction intermediates in electrochemistry. Now, let's suppose that we have an electrochemical reaction, like this simple reaction shown here, where we take iron porphyrin, electrochemically reduce it to iron 2, and with this complex, we will reduce oxygen molecule to 2 molecules of water. When we apply a potential to this reaction mixture, when we apply a little bit of positive potential, you can see the mass spectrum. We can oxidize the starting iron 3 porphyrin to iron 4 porphyrin. If we apply a negative potential, what you can see are the intermediates or putative intermediates from this reaction cycle. So we can see iron 4 hydroxyl, which is this intermediate. We can also see iron 5 complex that is basically derived from this reactive intermediate after it's connected to chloride and got protonated. And we can also see some species that we don't know exactly what they could be, but they look like some iron 3 porphyrin with incorporated O2. Now, this shows what mass spectrometry can give you. If you have high resolution, you know elemental composition. So you know that this iron contains porphyrin, and you know that it has hydroxyl. You could assume that hydroxyl is attached to iron, but you do not have any proof because you have just mass. So in this case, mass spectrometry is helpful, allows you to identify potential intermediates, but it's not a proof. You need to go further. So what you want to have is for each of this detected ion, some additional spectrum. And what we do is that for each of them, after mass selection, we measure infrared spectrum. So here you have infrared spectrum of iron 4 hydroxyporphyrin, and you can see that it has an OH vibration that you would expect for hydroxyl, and it has a characteristic vibrations of the porphyrin ring. Now, this is a simple case. If you go now to the degradation, where we did not know the structure in advance, if you measure infrared spectrum of this ion, you see that the spectrum is much more complicated. So the symmetry of the porphyrin had to be somehow broken. And when you do the theoretical calculation, you can assign this spectrum to the porphyrin that to the iron complex that has a broken porphyrin ring, and oxygen got incorporated into the porphyrin ring. Now, how does this work? I will simplify it. So you take any method that you have to transfer the intermediates from solution to the gas phase, or it can be also formed in the gas phase, or it can be also transferred from the surface to the gas phase. The ionization method does not matter. When you detect the ion, you then mass select it by some available mass spectrometry method, and the mass selected ions you transfer to a trap. In a trap, you have the ions and they are there for a certain time, and this time is available for irradiation with the laser, and this allows you to measure infrared or UV spectra. And I have this simple example. This is nitrine, triplet nitrine ion. And this is the measured infrared spectrum. What I want to stress here is that the spectrum is very well resolved, because you have basically single isolated ions in vacuum, and you measure their infrared spectra. So you do not have this broadening which you have in the solution. And also, there is no effect of the medium, so it's very easy to interpret these spectra by DFT calculations. And in the bottom, you see a DFT calculation of an IR spectrum of this ion, and you can see that there is a great agreement. In addition, we usually use isotopic labeling. So because these ions are mass selected, you can mass select an ion which has a sulfur 32 isotope, But sulfur has also naturally abundant 34 isotopes. So you can also one by one then select the other isotopologue, measure the infrared spectrum, and see which vibration bands shifted. And this you can model also in theory, and this will increase the confidence of the assignment. And I show you why it's important. Here, I just assume that the structure is triplet nitrine, but of course, it can be also different structure. It can be singlet nitrine, for example. But if you have a look at the predicted spectrum of singlet nitrine, it does not correspond to the experimental spectrum, because the shift of one of the SO vibrations would have to be found here and not in the position where we actually find it. You could also assume that the nitrine should rearrange and form much more stable isomer that is shown here. And you can see that it lies 150 kilojoules per mole lower in energy. So there will be a high driving force for this rearrangement. But spectroscopy allows you to see that this rearrangement did not happen. So it did not happen in solution, and it did not happen in the gas phase either. And you can see it, for example, here we have these three bands that are beautifully reproduced by the spectrum of triplet nitrine, but they are not present in the rearranged ion. Okay, so this is a method that really gives you confidence about the structure of the ions that you detect. I have also an example of spectroscopy in the visible range. So here's another nitrine now coordinated to copper. You can see that we can assign also its optical, its visible spectrum, electronic spectrum. Here is another example of a spectrum of complex metal hydride complexes that we studied. And I want to show here another advantage. Quite often when you study metal complexes, DFT calculation is not reliable in predicting ground state. So for this complex, for example, B3-lib method predicts a quartet state as the ground state, but it is formed actually in the doublet state. And you can see it by comparison of its experimental spectrum, which nicely agrees with the electronic spectrum of the doublet state. And since I'm here highlighting Dutch research and the possibilities that we have in the Netherlands, I want to mention that at our university, Radboud University in Nijmegen, we have a multi-user facility, which is called HFML Felix. And the Felix part stands for free electron laser that can be used for this spectroscopy that I have shown you. And it's a multi-user facility, so it's available for every one of you. You can apply for measuring time. They have extremely great equipment of mass spectrometers, so you can do a broad array of mass spectrometry experiments. And I want to just highlight a recent paper that they published, because I think it's really beautiful. So they have a connection of multi-imaging with infrared spectroscopy of mass selected ions. So you might know that nowadays it's available to do imaging of tissues with mass spectrometry techniques. You have a laser-based technique that ionizes species from the surface of some tissue, and you can for each point have a whole mass spectra, and then you can have a maps of different masses, how they are distributed over the tissue. So here you have an example of a mouse brain, some slice of mouse brain, and this is a distribution of glutathione molecule over this slice. Now, again, you don't know that it's glutathione, you just know the exact mass. But if you are doing this experiment at Felix, you can also connect it with measuring infrared spectrum of this ion, and then you can nicely assign these ions that are spread over this slice of brain are actually glutathione. So if you are interested in that, you can apply for measuring time, and you can also do experiments with your detected ions. Okay, so this brings me finally to electrocatalytic reactions. First, I want to explain how do we detect intermediates in electrocatalysis. So this cannot be done just by connecting simply the flask in which you are doing electrochemistry to a mass spectrometer, because the reactive intermediates are formed at the electrode. So they are not spread all over the reaction mixture, but they are usually connected locally to the electrode, and they have short lifetime. So you have to detect them from the electrode. So in order to do it, we developed a working electrode that we make from two parts. So this is made from carbon paper, but you can use also other materials. So we use two carbon papers, and in between we squeeze a capillary that is connected directly to a mass spectrometer. Now, again, we work with the reaction mixture under slight overpressure of nitrogen or other reactant gas. And this makes the reaction mixture slowly flowing through the working electrode to the mass spectrometer. Now, the dimensions are so that we are collecting sample at about 50 to 100 micrometers distance from the surface of the electrode. And this is exactly where the diffusion layer lies. So we sample the ions from the diffusion layer. Now, if I will show you how it works, I will show you first just calibration with ferrocene. So if you oxidize ferrocene to ferrocenium ion, you do cyclic voltammetry, you will see oxidation, formation of ferrocenium, and then you will have a drop of current because of diffusion limitation. And when you go back at certain point, you will again see reduction of ferrocenium to ferrocene. Now, when you study this reaction in the flask that is connected to mass spectrometer, you will measure this current, and simultaneously you will measure the abundance of this ion, ferrocenium, in a mass spectrum. And you can see that when we scan voltage in one direction, we can perfectly reproduce the onset of the formation of ferrocenium ion. When you go back, we see a little bit of memory. Therefore, we always scan just in one direction. Now, the advantage is that you can do it for all ions in mass spectrum. So here I had a mixture just with one ferrocene, but you can now mix different ferrocenes all together in one solution and measure their onset potentials simultaneously. And we did that, and also benchmarked it with standard voltammetry determined redox potentials. And you can see that this method is very reproducible. So we can determine the onset potentials of different species in one reaction mixture. And this allows us to study complex reaction mixtures and study what is getting oxidized, reduced, and when. And I will show it again on an example that you already saw in the outset when I was explaining the need for ion spectroscopy. So this reaction is oxygen reduction catalyzed by iron porphyrin, and it's mediated by electrochemistry. So when you look at the cyclic voltammetry measured for this reaction mixture, now we added ferrocene to have a reference inside. So you start to first oxidize, you oxidize ferrocene to ferrocenium ion, then you reduce ferrocenium to ferrocene, and then when you start to decrease the potential, at certain point you will see this current, and this current means that the reaction is running. So this current is current that you need for reducing oxygen to 2 molecules of water. Now, when you look at the mass spectra at certain points, when we start to oxidize, what is next to ferrocene, we also oxidize iron 3 porphyrin, so we can detect iron 4 porphyrin. Now, when we decrease potential to about here, you see the spectrum that you saw already once. So we can detect this intermediate, iron 4 hydroxyl, and others. Now, I will go back. You can follow the individual peaks, their abundance, as a function of potential. So now I will just look at individual peaks at all points of this cyclic voltammogram. So again, we start with ferrocene to determine 0, and then we are decreasing potential. Now this dotted line or dashed line determines the rate of the reaction. So the reaction is fastest when the red line has maximum. Okay, so when we look at the ions, you can see that the signal of iron 4 hydroxyl is maximum when there is a maximum rate of this catalysis. And this is exactly what you would expect, because the reaction is running and probably the last step is slowest. Therefore, we detect very selectively iron 4 hydroxyl complex. Now, what happens if we go to the yellow part? So we increase the rate because we increase the potential in the blue part of this voltammogram, but then we hit the maximum when we started to consume all protons that were in the diffusion layer. And because we don't have enough protons, the rate decreases. And what happens, or what we see in the mass spectrum, that we have an onset of a different process. And this process is degradation by oxidation. And this is because we don't have enough protons. So this step slows down. And instead of it, this superoxo complex sits in the reaction mixture and eventually goes to this degradation. Now, when we still increase the potential, at certain point, we hit conditions in which we do not have protons, but we also don't have any more oxygen in the diffusion layer. It means that not only this step slows down, but also the step of forming superoxo complex. And what happens is that these two species react together, they make mu peroxo complex, which finally degrades to mu oxo complex. This is final degradation process. So now I already showed you that we can, for all of the detected ions, we can do the spectroscopy and assign the structure. So we know that the degradation pathway leads to breaking of the porphyrin ring. We also know that this is a short-lived intermediate, and if you perform the electrolysis for longer time, you will see that this intermediate degrades to intermediate or to complex with a lower mass. And when we studied that complex, we found out that this is oxaporphyrin. So we know that under conditions that we do not have enough protons in the reaction mixture, we can degrade iron porphyrin to iron oxaporphyrin. And we detected many more ions, so I'll just quickly walk you through this reaction mechanism. And with this, I want to show how many details we can obtain from this analysis. So what we can see is in the catalytic cycle that after reducing iron to iron 2 porphyrin, it can react with oxygen to make a superoxo complex. Then it undergoes proton-coupled electron transfer to make a hydroperoxo complex, which after protonation at distal oxygen releases water, and we form a complex that is in biochemistry called compound 1. It's an extremely reactive complex, and we do not detect it as such. So it does not survive the transfer to a mass spectrometer, but we detect it indirectly after it attaches chloride and gets protonated as this iron 5 chlorohydroxyl complex, and this is a blue trace. And this complex undergoes another proton-coupled electron transfer and forms iron 4 hydroxyl, and this is complex that nicely reflects the rate of the reaction. Now, finally, this compound 1, detected as iron 5 complex, has another maximum in the situation where we are proton and oxygen limited. And when I will go back to it because there is another path to this complex. Okay, as I alluded to already previously, if you do not have enough protons, what happens is that this iron superoxo complex sits in the reaction mixture, eventually reacts with another highly oxidized complex there, and undergoes what we call Werther-Heme degradation pathways. It's a pathway that also happens in biochemistry. And under the conditions that the first step slows down, we have this dimerization pathway leading to iron mu peroxo complex, and this complex can undergo either heterolytic or homolytic cleavage. When it undergoes heterolytic cleavage, this is the way how we contribute again to the formation of compound 1, so iron 4 oxo complex with oxidized ligand, and therefore we see the second maximum under this diffusion limited complex in this trace. Now, these complexes then react with another iron porphyrin, because remember, this is already a situation that we are accumulating this complex in the reaction mixture, and therefore we can form one or the other mu oxo complexes that we detect under these diffusion limited conditions. Okay, so with this, I try to show you that mass spectrometry can have really detailed insight into reactions, and you can use this for studying organic reactions, organometallic reactions, but also electrochemical reactions. For all intermediates, you can do investigation of the structure by ion spectroscopy, and this coupling of electrochemistry and mass spectrometry allows us to study also mass transport effects at the electrode, and there are not so many methods with which you can study these details. So I think in future it can be really important for various applications, and we can study reaction mechanisms. Now, I think I don't have time for the story to show you another reaction mechanism, but I will just go over it. So this is an example where we applied it for unknown reaction, where our collaborators use cobalt complex to perform water oxidation. And during water oxidation, they use the cobalt intermediates to perform epoxidation reactions of alkenes. And again, I will jump over it because I don't have time to explain it. I will just conclude that with doing these mass spectrometry experiments using isotopic labeling, we could follow the whole catalytic cycle, characterize the reactive intermediates. We could also follow how the catalyst degrades, and we could also show that partially degraded catalyst is still active in the reaction. So we could get details that were impossible to get with other methods, because these complexes are not suitable for infrared or Raman spectroscopy that is usually used with electrochemistry for investigation of reaction intermediates. Okay, so with this, I'm close to conclusion. I just want to say that you can use these methods for investigating various reactions, and we have already shown it for several reaction mechanisms, and there are also other groups worldwide who are using these connections, and this can be an interesting tool to study reactions and improve your reactions. So what I did, I wanted to show you that with mass spectrometry, you can investigate intermediates in various reactions. I showed you some examples. I showed you that you can study kinetics. This is specific in electrochemistry, but you can do it for other reaction conditions in different ways, but it's always important to have a look at the kinetics. And mass spectrometry is an idea to look at side pathways and degradation of the catalyst, because this is usually minor pathways, minor problem, but it becomes a bigger problem if you are developing some reaction for longer time. So it's important to have a look at this, and mass spectrometry is an ideal tool to do it. So with this, I want to thank my group and you for the attention.
Thank you very much, Jana. You see the applause on your screen probably as well. Very insightful lecture. Actually, I'm very happy to be the host of this webinar because I learn a lot. The lecture is open for discussion. I look in the question and answer box, and there is a question from Nicola Bazarik, who asks, what is time resolution or minimum lifetime for the detection of an intermediate, let's say in a photoreaction by MS and IR? So about time resolution or the minimum lifetime.
So in photochemistry is of course critical because you will not be able to detect photoexcited species. They have too short lifetime, and for that there are different spectroscopic methods. But with mass spectrometry, you can study the relatively short-lived intermediates that are formed in photochemical reactions. And the shortest time that we got to is when you initiate the reactions already during formation of droplets. And then you can get to 10 milliseconds, 100 milliseconds of lifetime. If we are talking about transferring intermediates from solution to the gas phase, then they have to have lifetime in seconds and more, because otherwise they will not survive the transfer.
Okay, and Nicola says, thank you. So he's satisfied. I had a question myself about this diffusion layer around the electrode, because I can imagine that varies a lot depending on the supporting electrode and the solvent and so on. How difficult or critical is that, or is it always possible to tune that?
I mean, this is always unique conditions for different electrodes, for different electrolyte and different solvents. So you have to study it individually. But yeah, it changes the whole reaction basically always.
Yes, yeah, I can imagine. And anyway, now I'm asking this is, what do you do in this mass spec with this supporting electrolyte? Because you have to remove that, right?
No, so we use potassium-based electrolytes, potassium PF6 usually. You cannot use ammonium-based, so tetrabutyl ammonium-based electrolytes, because tetrabutyl ammonium is a very strong ion that makes ion suppression for everything else. So you would then see just tetrabutyl ammonium ion in your mass spectrum. But potassium is quite good. It makes clusters, so you can see one potassium, two potassium bound by PF6, etc. So you can see really large clusters, but they have defined masses, and in between you can detect your the species that you are interested in. And it has additional advantage. Potassium usually quite well tags neutral molecules, so it actually allows us to detect better neutral molecules than without the electrolyte.
I see. Yeah, because you only can see something that is charged, right? Yeah. Okay, thank you. And there are more thank yous in the question and answer session. And another question. In the case that the reaction forms isomeric structures, so the same M over Z ratio, can those be resolved during this technique?
Yeah, so you can use ion mobility to resolve them, and we use it as well. But unfortunately, ion mobility is not very friendly method for reactive intermediates, because ion mobility, the ions have to fly, so to say, fly in a gas, and then they collide, and they quite often you degrade them. So we use ion mobility only when we don't study very reactive ions. If we have very reactive ions, we have only ion spectroscopy, and then you can use so-called two-photon techniques. So you, with one absorption, you kill one isomer, and then you can study the other isomer. And in principle, you can do it one by one in mixtures. It's a complicated experiment, but you can resolve even more complex mixtures than mixture of two isomers. But it's time consuming, but it's possible.
Okay, Thank you. And also Michiel is satisfied with the answer. Okay. And then a final question, because of the time, by Marus. Are there other iron? And then it says 5, 6. So I'm not entirely sure about the question. So are there other iron, maybe 5 or 6 oxidation states or so?
So iron 5 oxidation state is it's reactive, but it's quite common. Iron 6 is usually not formed in the usual reaction, but you can generate such ions. But we never saw it in an electrochemical reaction or so. But you can generate them if you use specific ways to generate iron 6 complexes.
Hey, thank you. And with that, we thank Jana once again for her contribution. Thank you very much.
You.
And we go to the next speaker, and that is Siberen Otto from the University of Groningen. And he will talk about approaching the synthesis of life. Siberen.
Thanks, Adrie. Thanks also for the invitation to talk here. So I'm trying to bring up my screen. If things
I do
work
see your presentation
properly, then it will go full screen in a minute.
Right. Yep.
Yeah.
I see it full screen.
Good.
Yeah.
So what we're trying is, as many chemists, we're trying to make something, but we put ourselves to a really very large and maybe unsurmountable challenge, but let's see. And that's the synthesis of life. So if you like, the total synthesis of a minimal form of life. So we try to make things as easy as possible, but nevertheless. So I'm a physical organic chemist by training. So I'll also try in the rest of this talk to dive a little bit into the mechanisms by which fundamental aspects of life can emerge from mixtures of molecules. But yeah, so what we do fits into the bigger context of not only the synthesis of life, which for some reason actually remarkably few people are really active, but also maybe more peripherally, the question of the origin of life, which is a more established research field. So when trying to move from chemistry to biology, as you do when you want to start making a minimal form of life from really completely synthetic components, you have to change perspective a little bit from your upbringing and when you're at least brought up as a chemist. So rather than focusing on molecular structures, the focus shifts more to processes that molecules undergo. Rather than only looking at covalent bond formation, also the breakage of bonds becomes very important. And rather than only focusing on assembly, also disassembly processes really start to matter. And in particular, the interplay between covalent bond formation and breakage and assembly and disassembly is what you need to get to the state of life. It is indeed also what happens all the time inside anything that's living. Molecules are continuously being formed and broken, and they're assembling and disassembling. So if you really want to look at the real basic foundations of life, I think these are the 4 processes that you need to somehow bring together. So that then also gets you into the field of systems chemistry, where that interplay basically then defines the system. Now, in order to get to something that you can call a minimal form of life, you need to think about what characteristics that thing should have. While defining life is still very difficult, there's no accepted definition for life. One can perhaps agree, if not on a definition, then on a list of features, a list of characteristics that life should have. And these include compartmentalization, replication, metabolism. And once you have a system that has all these 3 aspects in it, it should be able to undergo Darwinian evolution. And in order for that to happen, you also need to maintain it far from equilibrium. So this is our basic plan. Can we see if we can bring these three aspects together into a combined system that is unable to undergo Darwinian evolution? And then we hope that evolution will complexify it further to a point that maybe one day it will become alive. So I'll touch on all of these aspects and their integration in the remainder of the talk. Let me start with how we started in this field, and that is with self-replicating molecules. And let me start with giving you the mechanism by which you can get to self-replication, although maybe in a way that's a bit different from how you used to think about self-replication or replication when it comes to what we see in biology with nucleic acids, DNA. So here's a bit different. Actually, looking at what you need to get to a simple form of self-replication, the ingredients are not very demanding. So there's a reversible oligomerization reaction. You take a building block, you allow it to oligomerize into oligomers of various sorts, but in a reversible way, so that the bonds between the monomer units can break again and reform with other partners. So if you have that and you have a self-assembly process of some of the oligomers you're making, so it's selective for some, then the assembly process will basically pull the material out of this reversible oligomerization reaction in a way that generates more of that specific material, and that gets you to self-replication. So this may sound a bit abstract, so let me try and make it a little bit more concrete by showing you a little animation of a system that starts very simple. It has one building block with two reactive ends in yellow that can combine to form different oligomers. In this case, these are rings that are being formed, rings of different sizes. It doesn't need to be rings. You could also think of the same thing with linear sequences. So the building blocks exchange between different rings, giving you the diversity in terms of different ring sizes. So you start with one building block and then you get a modestly diverse mixture of ring sizes. Once rings start to assemble, and that's about to happen now, two rings coming together, the assembly basically stabilizes the rings that make the assembly, and the assembly also pulls those rings out of the mixture that keeps replenishing them. And that gets you to a process of self-replication, where the assembly process drives effectively the synthesis of the ring that makes the assembly. Now, if you start to agitate mixtures like this, these long, thin assemblies can break. And by breaking, they double the number of ends they have from which they grow. And with that, the growth rate doubles. So this is a way to not only get to replication, but also to exponential replication. And this was, for quite a while, a big challenge in the field of self-replicating molecules, is how to achieve exponential growth. And this is rather important if you want to later go to Darwinian evolution. Right, so we have replicators working in this way. So let me recap how that system worked that you just saw. So we start off with a building block. I'm now going to fill in some of the chemical details as well. The building block now has two thiols that can be oxidized to make disulfide bonds, making the rings, connecting the building blocks within the rings. Disulfides are reversible bonds in the presence of thiols, so you have exchange between different ring sizes, a relatively slow nucleation phase, but once that those nuclei start to grow, you enter a growth, if they're long enough, a growth breakage cycle that allows the exponential replication of the rings and the fibers that they're forming. What I did not show in the animation, because we've discovered it after the animation was made, is that these fibers can also bind the precursors from which they're made upon replication to their sites. So you have reservoirs of precursors accumulating on the fiber sites, and that helps in the process of replication. And it also has some consequences later on in my presentation. So please remember this accumulation of precursors to the replicator on the sites of these replicators. So I told you that the building blocks contain thiols, and the entire structure of the building block looks like this. So it has an aromatic ring with the two thiols and then a bit of peptide attached. Peptide was designed to have hydrophobic and hydrophilic residues that alternate. And with sequences like that, there's an inherent tendency to form beta sheets. But beta sheet formation usually requires a longer peptide than this short guy here. So beta sheet formation doesn't happen with the monomer. It also doesn't happen with small rings, but if you get a large enough ring, there's enough sort of stickiness to the peptides, or enough of these peptides that then individually want to make beta sheets, that you do now get beta sheet formation. So multivalency kicks in. So beyond a certain critical ring size, or at a certain critical ring size, that particular ring then assembles and starts to replicate. Right, so now you know how replication works. Let's see if we can integrate it with a very simple form of metabolism. Now, metabolism, as we all know, requires catalysis. So the central question now is, how can replicators acquire catalytic activity? And it turns out that also that emerged in the process of assembly, because assembly of a peptide like this puts a lot of lysine residues together close in space. Now, lysines normally would like to be protonated at neutral pH in water, where we conduct our experiments. But if you put a lot together, having them all protonated would cause way too much charge repulsion. So actually, you have a lot of lysines now in the assembly that remain unprotonated, whereas in the non-assembled state they were. So the effective pKa, if you like, of the lysines shifts by about 3 units. So it's a substantial effect. Rendering these amines of the lysines free for doing catalysis. On the same fibers, there are also hydrophobic residues that can act and shape hydrophobic binding sites on the fibers for substrates, but also for cofactors that can be used to mediate catalysis. So it turns out that with one and the same peptide sequence of 5 amino acids, we can catalyze with these fibers a remarkably diverse set of reactions. So here's the current list, and it continues to grow. So with one and the same peptide sequence in the fibers, we can catalyze FMOC cleavage, cofactor-mediated photoredox catalysis, but also retro-aldol reactions, Knoevenagel condensation, hydrazone formation, ester hydrolysis. I'll not have time to touch on all of these. I'll only go into a bit more detail on the first two. So FMOC cleavage, the way that works, for normally to cleave an FMOC group of something, you need a base to attract the proton here. And normally FMOC groups are deprotected under reasonably basic conditions. But in the presence of our fibers, we can do this even at neutral pH within minutes, because the fibers have these basic amines that are non-protonated at neutral pH because of the reasons I just explained. So those act as Brunstad bases, and probably the protonated lysines play a role as well in helping with the further fragmentation of the FMOC group, the decarboxylation, and in this case, liberating glycine. Now, this is catalysis, but not necessarily metabolism, because the glycine is of no use to the system. But you can also make catalyze a reaction that is useful for the system, to the point that it accelerates its own replication, and that is done with the mediated singlet oxygen production through a cofactor that's being bound to the replicator. So we can use a range of different cofactors that, upon binding to the replicator fiber, become more efficient at producing singlet oxygen than they are in their unbound state. And the singlet oxygen then accelerates the conversion of thiols to disulfides. So it converts the building block to the small rings, and these small rings are what the replicator needs to replicate from. So in this sense, the replicator effectively produces its own precursors. It basically produces the molecules it needs. So you can regard that as a simple form of metabolism. Right, so that is combining replication with metabolism. Let me now progress on to the topic of Darwinian evolution. also of replicators that are able to do reactions. So we've done a few things in this area recently. I don't have time to touch on all of this. So we've developed a system where the replicators change their own environment through again this photoredox catalysis. The oxidation state of their environment changes, and that affects the competition between different replicators. And one can consider that as a very simple sort of eco-evolutionary dynamics. Replicator changes its environment, and environment affects the competition between replicators. I will not have time to say much more about it. We also worked on what's the essence of Darwinian evolution, the idea of competitive exclusion, that is survival of the fittest. So if two replicators compete for the same resource, then only the fittest should survive. And we've been able to show that that also holds, that principle from Darwinian evolution holds for very simple abiotic self-replicating molecules. What I do want to spend a little bit more time on is selection for metabolic activity. So I just showed you how you can get a very simple sort of metabolism. Can you now use Darwinian evolution to select specifically those replicators that are a little bit better at doing this metabolism than their competitors? So the idea is now to make a replicator based from not only one, but two different building blocks, which would generate either a set of replicator mutants that differ in how many of these building blocks they incorporate, or maybe two different replicators altogether if they one decides to to be formed from one building block and the other from the other. I'll show you both examples. And the idea is that these replicators again can generate the singlet oxygen upon binding a dye, where the singlet oxygen helps converting the building blocks into the precursors that then bind to these replicative fibers and are there converted to more replicator. So that's the basic chemistry. Now, in order to do evolutionary experiments, you need to set the system up so that there's not only replication, but also some element of death. And one of the simplest ways of implementing that is by putting the replicators in a flow system, where you continuously supply them with the building blocks they need to replicate from, and also the cofactor that is needed for the singlet oxygen generation. So they're being flown in, but at the same time, at the same flow rate, you also flow out part of the solution. And that then amounts to death. If you're no longer in the vial, then you can no longer do anything. So if you're a replicator in here, you better replicate faster than you're being flown out, or at least as fast. Otherwise, you'll go extinct. So that's the setup, so not very complicated. So let's see what happens. So we designed two different systems. One that gives rise to a set of mutant hexamer replicators, and mutants in the sense that they can incorporate different ratio the two building blocks we start from, and the two building blocks are very similar to what I showed you before. They only differ in here. This one has a phenyl ring, and that one has a naphthyl ring. For the rest, they're the same. So in this system, you make mixed hexamer replicators. So the red building block is the naphthalene, the blue is the phenylalanine. And the idea was that if you then subject that to this evolutionary experiment, that should select for those mutants that are best at catalyzing or the formation of singlet oxygen. So that's system one. In system two, we again mix two building blocks. One is almost the same as this one. The other one has a reversed peptide sequence. So here you start with hydrophobic, hydrophilic, hydrophobic, hydrophilic, and this is just the opposite. It's hydrophilic, hydrophobic, hydrophilic, hydrophobic. So these guys don't make a very good beta sheet between them, but they can do this with themselves. So they then have a tendency to self-sort. So you get the red guys predominantly in trimers and the blue guys predominantly in hexamers. And then you can compete these against one another and see if through evolution, one actually wins or has a benefit or has an advantage over the other. So let's see what happens. So in the case of the hexamer systems, if you don't do any evolution, you end up with a more or less statistical distribution. But if you start evolving, then you see this distribution shifts and you make more of the replicators that are richer in the phenylalanine building block. And we've then been able to confirm that these guys, those mutants, are indeed also the ones that are more active at generating singlet oxygen. So it's a modest shift, but it's the first example of Darwinian evolution outside the context of life or biomolecules in chemistry. For the trimer-hexamer competition, evolution also has an effect. You see here what happens prior to evolution. You have a small amount of hexamers, more trimers. So the trimers dominate the population, but after running this in a slow setup, the hexamers are now more prevalent, and the trimers less so. So again, we see that evolution induces a shift in the replicator population. So this seems to work, but it's actually interesting to ask the question, why? And it's even not entirely obvious that it should work. So in order for Darwinian evolution to select for catalytically active species, the most active replicators should be the ones that also benefit from that activity. And that, in a homogeneous solution, is not a given. You could imagine if some replicator catalyzes something and that's then liberated into solution, all the other replicators, also the ones that do not catalyze anything, can eat that material and grow from it. So the fact that in homogeneous solution we still get some, as you saw in the previous slide, there maybe not massive shifts, but some shifts in population, it must mean that there is something special going on. And it turns out that this system has a number of features that allow for this selection to occur, in that the cofactor is located on the replicator, so the cofactors are not free in solution, and the cofactors are that generate the singlet oxygen. Also, the singlet oxygen doesn't distribute homogeneously through solution because its lifetime is too short to make it to neighboring replicators. So basically, if the singlet oxygen is generated, it will only act very locally near to the replicator that created it, and then the singlet oxygen makes basically the food for the replicator, and the replicators, as I showed you before, have a tendency to bind that food. And we've done some controls to show that the exchange of food between different replicators is also slower than the replication process. So replication is faster than food exchange. So that means all these features come together to ensure that kleptoparasites, so these would be the replicators that don't do catalysis, but kind of steal the food from the ones that do, but kleptoparasites in this system have no chance of, or at least a reduced chance of survival. But as you can imagine, for this reaction, this works, but do we now have a general way of selecting for catalytic activity by Darwinian evolution? Well, no. But how can we make it general? The solution there is compartmentalization. So we may remember if our problem is kleptoparasites, we may remember from the corona pandemic when we had to deal with parasites, viruses, that the solution is is locked down, so you lock yourself up, which is basically a form of compartmentalization that prevents spreading of parasites. So we need compartments if we're to get better at doing Darwinian selecting for better catalysts by Darwinian evolution. So let's then look at how we can integrate compartmentalization with replication and metabolism. Now Darwinian evolution is in gray because we haven't got to the point of evolving a system that has this ternary integration, but at least we succeeded in the ternary integration very recently. And for the compartments, we chose coacervates. Now coacervates are funny phase-separated systems. If you haven't come across them, they're formed by, usually the textbook example is a cationic and an anionic polymer. If you put them in water, they want to phase separate because of charge neutralization. Oftentimes this results in just precipitation, but there's a subset that are kind of frustrated. They want to phase separate, but they can't quite get the charges aligned and organized nicely in its own neat phase, and then they get sort of arrested halfway, where there's still a lot of interaction to drive partial phase separation, but there's also still a lot of interaction with water. That means that we form water-rich droplets in a continuous water phase. So that's counterintuitive, but this is the way it works. It has no membrane barrier, so unlike vesicles, you can move things in and out relatively easily. Because of this frustrated nature of interactions that are only partially formed, it likes to still interact with lots of stuff. So hydrophobic molecules, but also charged molecules, tend to concentrate up inside coacervates. So that's also really all very nice features. They have only one less nice feature in that they want to spontaneously coalesce. So they start off as droplets, but if you don't do anything, after a little while, all the droplets fuse and you have two separate phases. But still, they look like attractive things to integrate into our systems. So we decided to design, this is work by Armin Kiani, who came up with this design, a dithiol building block like our replicators, but now it has a peptide that is a di block, cationic and an anionic block, that form the charge attraction with themselves, basically. But again, the building block itself doesn't form a coacervate, but if you make them into rings, then they become sufficiently sticky that at some ring size, indeed, a phase separation into coacervates occurs. It happens with this system to be tetrameric rings that do this. So here's some data that shows what happens when you take this monomer and you allow it to oxidize. You lose the monomer in the beginning when they're still where you're below the critical aggregation concentration for coacervate formation. You get pretty much all the rings. But the moment coacervates start forming, you see that you no longer get additional trimer, but you only make more tetramer. That shows that indeed here it's the tetramer that wants to make these coacervates. So nice, we can make coacervates. Can we couple it to replication? Well, remember we had these photoredox catalysis of singlet oxygen production that allows us to convert thiols to disulfides. That also works with the coacervate forming thiol, no surprise. So putting a replicator, the cofactor, light, and just regular oxygen enhances this rate at which these coacervate forming molecules are formed. And also not as a surprise to us, once you form the coacervates, the replicator spontaneously partitions into it. So here's some data to show that this coacervate formation with a replicator, dye, and light goes a lot faster than in a control experiment. And here's a picture showing the coacervate droplets and with a labeled replicator to show that the replicator is indeed inside, and you can quantify this and work out that you're about 800 times more concentrated in replicator inside than outside, and also that replicators have a very slow rate of exchange between droplets, which is also rather important for droplets to maintain their own, if you like, genetic identity. So replicators, once they're inside, they still continue.
Sybren, you're running out of time, so could you?
Okay. Yeah, I'll speed up so they can make coacervates grow, and they can even make them divide. And for that, we again use the replicator ability to catalyze reactions. So you've seen already that you can make building blocks into coacervate material. In order to make replicators, the coacervates divide, theory suggests you should also be able to do the reverse, and namely destroy again the coacervate materials so to destabilize them. If you try this with a reducing agent you add externally, you can make droplets shrink but not divide. If you do it with the same reducing agent, but you let it be produced by the replicator inside the coacervate, then the droplets do divide. So here's the FMO cleavage reaction that I introduced you in the beginning coming in. So we protect our reducing agent, so this molecule here is in the middle with FMO groups. And then in the presence of the replicator, these are cleaved, liberating the reducing agent, and then you get indeed from big droplets after a while to lots of small droplets. So with that, I think we've made another rather big step in the direction of the synthetic form of life, and by integrating these three fundamental aspects, we have not yet done any evolution on this system, but that's going to be soon. And then the question is, where does evolution take us? And this, I think, is the biggest hurdle, not only to our work, but also to, I think, the origin of life, is to how can anything primitive that's capable of evolution become access open-ended evolution, where there's a continuous space for more evolutionary inventions. We don't know how to do that. I don't think anyone knows how to do that, but I think that's the real big question that's still out there. I should stop here, Adi already mentioned, but not before thanking the people that did the work. So this is the group at various times. This is the latest picture in our new building. Here are labs. This is where we have coffee. We're very happy there. Very happy with the people that fund us. And if you come, if anybody is interested in joining the group, there are some open positions right now. And sorry for taking a little bit more of your time. Thanks for listening.
Thank you very much, Sybren. Well spent time. I enjoyed the lecture very much, and the lecture is open for discussion. I had a question about the fact that this supramolecular aggregates, so these piles of molecules, they do catalyze reactions. That looks to me very much like enzyme catalysis, but enzymes are, yeah, sometimes also supramolecular, but at least they are mostly one peptide chain. Is there a closer link between those?
Well, I think they are, our systems are assemblies, but they're pretty sturdy assemblies in the sense that once you're in the middle of one of these stacks, you don't get out again as a ring. So in that sense, they're pretty fixed there. So maybe from that point of view, it's not so dissimilar to an enzyme, except that I think I mean, enzymes are really have very, very well defined, basically active sites. I think these fibers are much more disorganized, and this is probably what makes them so versatile. In that, I mean, we've done some experiments with at least the retroaldo reaction that show that the number of lysines, so lysines are important for that reaction, the number of lysines that are actually doing it is far less than 10% of the total. So that means that along this heterogeneous fiber assembly, there are some sites that are good. And for this particular reaction, and our hypothesis is that the fact that we can do so many reactions is that there are other sites that are then actually good for other reactions. So that is, I think, where it becomes very dissimilar to an enzyme, because that usually has one specific thing, and there are good evolutionary reasons for that. We're dealing with a much more poorly controlled set of different sites on the same fiber, presumably.
Yeah, thank you. I see here a question from Wim Nijskens. Is the reason that you're forming four-membered rings in the coacervates that they're combined monomers or oligomers to the side? And in other words, there is no space for that in six-membered rings. Do you get the question?
Yeah, but I think the combining on the side was with the replicator. We don't need a replicator to form the coacervate. So the coacervates also don't form fibers. They're just rather loosely attached four-membered rings. So I think the four-membered ring formation is because that's giving enough interaction energy to hold the things together. Plus, we've done some molecular dynamic simulations that show that they also tend to fold up in a certain way. So the four-membered ring is not a completely cyclic open one, but it kind of collapses. And that might also help to stabilize the system, so you have less interaction of water with these hydrophobic aromatic rings with the thiols on. But it's a bit hand-wavy explanation. We don't completely understand why 4 and not 5 or 3.
Yeah. Okay, thank you. In view of the time, we thank Sybren once again for his contribution. And we go to the next speaker, and that is Professor Kim Bonger from Leiden University. And she will lecture on chemoenzymatic and metabolic protein labeling strategies. Kim. Yeah, I see your presentation already. Yeah, there it is. Thank you.
All good?
Yeah.
Perfect.
Looks very good.
Thank you, Adri, for the kind introduction, and also very happy to be part of this lineup of speakers. Very nice to give an overview of what's happening in the Netherlands. Yeah, so I'm trained as a synthetic organic chemist, but we've moved pretty much into the field of biology and immunology. I moved last year from Radboud University to Leiden University in this very new building. And a lot of the work has been also performed at Radboud University and now continuing at Leiden. So our lab interested in a lot of tool development and chemical biology strategies in order to understand immune diseases. We have been developing a lot of bioorthogonal chemistries in the past. We're focusing on new reactive species that we can use for biomolecule labeling. And we use these in order to have more specific targeted drug delivery approaches. And also we have a lot of interest in understanding the biological processes that play a role in immune disease. And therefore we develop a lot of activity-based probes for monitoring the enzymes that are involved in these processes. So I have two major topics for you. One is on nitrogen ion labeling approaches, and we apply that in the targeted drug delivery involved in autoimmune diseases. So I will cover these two topics also because it's a very big focus of our lab in the current or coming couple of years. So nitrogen ion labeling. So I was interested or became interested in these species. I was not very familiar with that, especially not in biological contexts. But when I was looking in literature a couple of years ago, I found, well, not this article, but at least that anilines, so arylamines, they are very toxic species. That was already recognized 130 years ago. That people who were working in a fuchsin factory, so this is the violet dye, so and that's made from arylamines, and these people, they have a very high occurrence of bladder cancer. That was stated by this Dr. Rijn in 130 years ago. So a couple of years later, it became apparent that the toxicity of anilines is very much dependent on what type of specific enzyme people express. So especially arylamine and acetyltransferase. So we call it NOT for short. And what happens in the body is that you have arylamines. They get oxidized by P450 enzymes. And you have this enzyme NOT that can acetylate this species either on the nitrogen or on the oxygen. And what happens if it acetylates on the oxygen, you will have this heterolytic cleavage of the acetyl-oxy function, and you end up with this nitrogen ion. And this is a very reactive species. It lives only for a couple of femtoseconds, and it's a very electrophilic species that can interact or react with nucleophilic species, especially in DNA and RNA, but also in proteins and other small molecules that are present in cells. So you can imagine if you have this nucleophile being your DNA, you introduce a mutation and you have a high occurrence of cancer. So I thought this process was really interesting, and maybe we can use it also for more chemical biology approaches. And what appeared is that NOT, so this enzyme, it can also undergo an NO-acetyl transfer mechanism where the acetyl from this hydroxamic acid is transferred from the nitrogen to the oxygen. So you can have an enzyme-induced formation of this well, nitrogen radical, basically. And our first instance that we want to explore this enzyme is in proximity labeling. And for those who are not familiar with proximity labeling, it's a very common technique used by a lot of biologists that are interested in the surroundings of a particular enzyme in the cell, for example. So what you do if you have a protein of interest, you fuse it to an enzyme. This enzyme can convert a in-reactive species into a very reactive species. This will diffuse from the enzyme site and it will label neighboring proteins. And because we have this very occurring mechanism, we can add, in our idea, an in-reactive hydroxamic acid species, and which is then converted into this nitronium ion and labeling proximal proteins. So 2 of these labeling methods have already been published a couple of years ago. They are called APEX. This is based on an enzyme that converts a phenol into a phenyl radical, and this reacts with proximal proteins. But this proximity labeling method requires millimolar concentrations of hydrogen peroxide. And this is, of course, for lifestyle imaging or just cell purposes, not very suitable. And then there is another method which is based on a biotin ligase. This requires biotin. It activates it into a biotin AMP, and that species is a very electrophilic, reacts with proximal lysines on proteins, and you have biotinylation. And these proteins you can then purify from your cell mix and identify what are the proximal proteins. So both these methods had benefits and drawbacks. So here you have the hydrogen peroxide, but biotin is also present in your cells. So it's very hard to discriminate between the originally biotinylated proteins versus the ones that you induced. So we thought we could complement the strategies using this proximity labeling based on NOT. And for that, we can make hydroxamic acids in lab, and we functionalize it with all kinds of, well, molecules that we are interested in, either biotin, but also fluorophores, or just biotoggle reactive handles like azides or alkynes or whatever you wish. And you can isolate, or you can use these handles for visualization, isolation, and analysis of the proximal proteins. So already a long time ago, students start to prepare the compounds. So in this case, the hydroxamic acid probe, and we also have the amide as a control. We have a general cell line, so it's a hex cell line or 3-3, doesn't matter. You can transfect these cell lines with the NOT enzyme. And in this case, we used a NOT enzyme that has a nuclear localization signal. So the enzyme is localized to the nucleus. And upon addition of the probe, you then expect only the nuclear proteins to be labeled with our compounds. And for readout, in this case, we have a fluorophore, a FITC dye, to see how well our method works. So here you see just some cells that they are stained with a DNA stain, transfected with the enzyme, and you see in red, so this is also a tag it has attached to it, these enzymes are expressed. And indeed, if we have our probes added to the cells, we see only cells labeled that have on the NET enzyme as well. And the control probes, they don't have any labeling. So this was very promising. We can also do it the other way around, where we translocate NOT to the cytosol and export it from the nucleus. You see there's nice labeling only in the compartments that we have the enzyme in. So this was very promising. It was also very fast. We saw very fast labeling. But we also found something very interesting later on, and that is that the labeling was very much dependent on the type of probe that we used. So we made different types of hydroxamic acids based on different substituents on the aromatic ring. In this case, we have an ether function. It has very nice co-localization. The control compound lacking this oxygen does not have any labeling. But if we go to some other hydroxamic acid species, so less electron rich in this case, we saw very diffuse labeling, and we did not see any co-localization. And in this case, we have even, well, more electron withdrawing group on that position, and we saw completely absence of labeling. So this was very interesting, because now we can have, depending on the probes that we add to the cells, we can tune the radius of labeling, or we can use the enzyme also for different applications. So our hypothesis why this different substituents give such a different labeling pattern is because if you look at the mechanism, first you form this acyloxyarylamine. This is a rather stable species. So, well, it is unstable, but rather stable compared to the natridium ion. It can live microseconds, milliseconds. And we believe that if you have this substituent on the para position, you can tune this stability of the acyloxyarylamine intermediate, where electron-rich substituents, it makes it very unstable. You get very fast cleavage, and electron-poor substituents, you may end up with a very stable species. So if you have a very electron-rich, you can end up with very fast natridium ion formation and proximal labeling of the NAT enzyme. And the other way around, when you have electron-poor substituents, it allows the compound to basically fuse away or diffuse away from the enzyme, and you can tune the reactivity and labeling of other substituents. So can we use that knowledge? We thought it would be very nice if we can use this in this enzyme as an in case of an antibody-directed enzyme product therapy. So what is this? This is a strategy where you can fuse an enzyme to an antibody, and antibodies are directed, you can very specifically direct it to a specific cell or antigen that you're interested in. And you can fuse this engineered enzyme to the antibody. And then if you add a particular drug that is caged and thereby not toxic, it gets activated by the enzyme, and the toxic drug is only released on the place that you have your enzyme targeted to. So as a proof of concept, does this work? So we have a toxin here in red, just a toxic molecule, and we cage it with this hydroxamic acid. And we know that this benzylic alcohol, it's not that reactive. So it can diffuse away from the enzyme. In this case, if it's caged, our cells would be alive if we add it. Although if we add our NAT enzyme, you will have the transfer of the acetyl, the formation of the natrinium ion, and then a nucleophile will attack on one of the positions on the ring, giving this substituted arylamine. And this is a very common self-eliminating linker that's used in product therapies, this amino benzyl oxy. And you have a self-eliminating cascade releasing CO2 and then your cargo drug. So to see if this works, we synthesize a model substrate. So this is an arylamine paranitro substituted. If it's caged, you do not see any color. But if it's released, you release nitroaniline, and you see this has an absorption of 385. You can measure this. So we did our first experiments, normalized what we saw. We added our prodrug and the NAT enzyme, and we saw drug release, nice, but it would never add to the what we would expect. So there's always a gap between the observed color that we would expect if we do not have the cage on there. And that can mean several things. Either our enzyme is not active, so we tested that by adding more enzyme, but it doesn't really help. Or there is something else going on. For example, we have enzyme inhibition, or there is the compound is just not fully released. But this was also not the case, because when we add another batch of the substrate, we have increased release again, but still not completely to the full expect we had. And then we looked a little bit further into the mechanism, and we thought like, okay, if we have this cascade going on, you have, of course, all these isomers and tautomers of the positive charge, and the nucleophile can attack on these positions. In this case, you will have drug release, but if you have the nucleophilic addition, in this case, I added water, but this can also be other biomolecules, you will have a species that will not allow release, and you end up with this strategy, with this structure here. So we are very excited about this enzyme-mediated natrinium ion formation, but also on different ways of generating natrinium ions. We believe that we can use them for bioconjugations and modifications, but also for targeted drug delivery. And we have now a lot of manpower to finally figure out how exactly the rules are in this deuterium ion chemistry in biological conditions, and especially because they have very unique reactivity towards biomolecules compared to water. And also we aim to tune this reactivity. So we have now prepared a lot of different substrates where you can see if we can tune this reactivity to more self-labeling, but also to more diffuse labeling and drug delivery applications. And especially, that's my bridge to the second part of the talk, we aim to use this for targeted drug delivery, either via the ADAPT strategy, so the antibody-directed enzyme product strategy, or cell engineering strategies. And we're especially interested in autoimmune disease, rheumatoid arthritis. Rheumatoid arthritis, many of you might know that it's a very painful disease. It's an autoimmune disease, so your body attacks yourself, and it results in very painful joints and misformed joints. And the basic mechanism behind this disease is it causes autoreactive immune cells to produce a lot of antibodies that are directed against a particular post-translational modification called citrulline. And citrulline is a post-translational modification that's coordinated by PAT enzymes from arginines. So people who have very high amounts of these antibodies, they get inflammation of these joints, and they're called ACPAs. And these ACPAs are produced from plasma cells, and these are derived from B cells. And it appeared that people who have very severe autoimmune disease, if you deplete the B cells, that it has a very positive outcome. So rituximab, it's an antibody that's currently on the market, and it completely depletes all the B cells. And it is used for people with severe autoimmune disease. And it works nicely, but it also has selectivity issues because you completely destroy your whole B cell compartment. And that means that you're also very sensitive to, for example, infections or other triggers that you have built your immune system or trained your immune system throughout your life. So we were very interested in designing a antigen selective precision therapy towards B cells, but only the B cells that cause disease and not the B cells that you would like to keep. So we teamed up already more than 10 years ago with collaborators at the Leiden University Medical Center, René Toes and Uri Scherer. They are rheumatologists. They said, can we just not make a very specific therapy for autoreactive B cells? And the one unique feature of autoreactive B cells versus the other ones is the B cell receptor. And this is exactly the receptor that recognizes this antigen. And we thought at that time very naively that it would be very nice to do so because the field of antibody drug therapies have become very big and popular and on the market. So it would be very easy to transform this into an antibody or an antigen selective B cell therapy. So all we needed to do is to make an antigen that's recognized by the B cell receptor in patients with autoimmune disease, in this case RA, and we connect it to a drug, including a linker, and that linker is then, if it's recognized by the B cell receptor, internalized, cleaved by enzymes, and then you can release a specific cargo, like a drug or something else, that can then kill the cell. And well, it was not that easy as we thought it was. It's been a long stretch. We started with, we need a target antigen, and that was for autoimmune disease in RA. That was present. It's called cyclic citrullinated peptides. It's this structure here. We can easily synthesize it on solid phase chemistry, cyclize the compound because it has a single cysteine here that we can use for cyclization. And we introduced a lysine molecule on the beginning that we can use for modifications, in this case, a biotin. We always have the citrulline, which is over here, that is recognized by a large patient population. And very interesting, if you change it to an arginine, so just one single atom replacement, it's completely absent in recognition. So this is our target peptide, and we explored whether we can use it for targeting the B cells. And we were especially interested in the affinity, but also cell activation, and especially the internalization. So we made different constructs based on the monomer, a dimer, but also a more bigger constructs. And I'll show you how they end up looking. So here is the peptide, and we can fuse it with either an azide or a biotin or a fluorophore for visualization. We made a monomer, and if we introduce an azide there, we can make a dimer. So we just click it with copper catalyzed click reaction to this triazine core, and this triazine core then we can fuse also with a biotin or a color. And if we have a biotin on these monomer and dimer, we incubate with streptavidin, and you get a tetramer and an octamer. So we looked at the binding affinities of all these compounds, and we saw a very nice binding, so very low nanomolar affinity, but you really needed a multivalent construct, so dimer, tetramer, or an octamer. And the monomer has millimolar affinity, which is not sufficient for efficient targeting. We also saw that the activation was also dependent on the clustering of the receptors, and that means that you need this multivalency in order to get internalization. And what you see here is a cell, single cell, and all the red dots are the constructs that are actually taken up inside the cell. And this is what you want for targeted drug delivery. So we have this very nice construct. Everything was in order. And we made a construct, the dimer, and we fused now a toxin to that, because these are the toxins that are already on the market for antibody drug conjugates. And they fuse a toxin MMAE. This is an inhibitor for cell division. It has a cleavable linker that allows release of the toxin when it's entering the cells. And here are our two peptides targeting CCP peptides. And we evaluated it on our cell line. So this is the 3F3 cell line. Not very important, but what is important, we saw cell killing, but at a very high concentration, and it was not selective for our targeting cells versus a non-related cell line. So we were very disappointed. It took us a lot of synthesis, a lot of figuring out how these peptides bind and behave in cells. But then we were thinking, why doesn't it work? Because if we use it on an antibody, so this is an antibody targeting the same B cell receptor with the same toxin, we saw very efficient cell killing, which was very specific for, or not specific for the B cell receptor, but we saw very strong cell killing, almost as strong as if you have just a free toxin attached to it. And we also saw that if you deplete the B cell receptor from a target cell, so this is a knockout, we saw a complete absence of the killing. So it is B cell receptor mediated. So why do they not die? And we were thinking many different reasons, but I think the most obvious in the end was the amount of toxin that we can load. So we have 2 antigens and 1 toxin. And in antibody drug therapies, you often have multiple toxins on the drug. So for our compounds, it was not possible to even go further because the targeting molecules and the dimers, they ended up being, having bad or unfavorable physical-chemical properties like insoluble compounds, etc. So we switched to a different system, and that was a system on polyisocyanopeptides. And this is a system, it's a semi-flexible polymer, it's bio-based and very hydrophilic. And it was developed at Radboud University in 2013. And the nice thing of these polymers is, depending on the side chain of the polyethylene glycols, as well as the catalyst that you add, you can have different types of polymers and fibers. And in this case, we end up with fibers that are about half or 500 nanometers or 250, 500 nanometers in length. And the nice thing in these polymers is that you can introduce monomers that carry an A side. And these A sites you can use in bioorthogonal click chemistry in order to functionalize them. So in this case, we made PIC polymers carrying these antigens that we have to increase the amount of targeting, but also allowing us to increase the amount of toxin that is, or relative toxin, I have to say, that is present. So they bind similarly as our dimers, so that was very happy to see. And they also have very good internalization because they are very more in length. We were almost afraid, like, do they internalize or can they internalize? But they internalize very nicely. And now we can also introduce increasing amount of toxin molecules relative to the targeting molecules. And we calculated that if you have 5 equivalents compared to the targeting, this would be the same affinity, or the same toxin cargo amount compared to the antibody. If we have half of them, to see what would be the effect. And indeed, if we now look at cell killing, we could finally kill cells in a very nice low nanomolar CCP concentration. And if you also compare it with cells that are not related, so other targeting cells, these cells, they are not sensitive to these kind of structures. So in this way, we are now pursuing this for in vivo experiments. They're actually going on as we speak to see if we have targeted drug delivery also in vivo. And we also pursue other strategies where we can either have the targeting, but also engagement of other receptors that are present on B cells in order to attenuate B cell receptor signaling. The last couple of minutes, I think I still have a couple of minutes left. I want to show you how we are pursuing this antigen selective therapy, because we're very interested in expanding this. And we recently switched to T cells. And T cells are the cells in your body. Basically, they are guarding your body and they kill anything that is not supposed to be there in your adaptive immunity. And chimeric antigen receptor T cell therapy, it's been one of the breakthroughs in the last couple of years, not only for autoimmune disease, but also especially for tumor targeting. And what the therapy entails is that if you have a patient, blood is drawn from a patient, T cells are isolated and grown, and then transduced with an artificial T cell that can target your tumor cell or target cell that you're interested in. So we were very interested in seeing if we can employ this T cell therapy to target autoimmune disease, and especially in RA. But as many autoimmune disease, as you may recall from the introduction, they are recognizing often non-naturally occurring modifications, such as citrulline. And what is now the problem is that you cannot genetically encode citrulline. So we have to come up with something else. And this is where synthetic organic chemistry will come into play. So instead of transducing an artificial T cell receptor, which has a genetic encoding targeting region, we now introduce a SNAP tag. And this SNAP tag is a self-labeling protein that's engineered already a couple of years ago. It allows the covalent modification of the benzyl guanine tag. So if you add a synthetic molecule to this SNAP tag, you have a covalent modification of the protein. So we synthesized again our targeting peptide, have the labeled protein, and here we have a fluorophore to see if we can actually use this synthesis or this scheme for for labeling and targeting of our artificial T cell, synthetic T cell. And we can, so here you see nice labeling of the T cell, which is dose dependent. And if we do a co-incubation, see I have to speed up a little bit, with our targeting cell, then we see very nice engagement of our B cell, this is now here in green, and our targeting cell, which is now here in red. And we can use synthetic organic chemistry in order to engage these cells together. And we can also have cell killing of this green autoreactive B cell by the T cell that we fluorescently and targeted labeling. And if you have cells that are not expressing our antigen, then T cells won't recognize it and will just happily leave them untouched. So where are we going with this? The T cell community, it's very promising, but it also has a very high risk of a side effect called the cytokine release storm. And it can actually, it happens already in 70–80% of the patients that receive CAR T therapies. And it can also cause potential death. And several clinical trials have to put on hold because there were so severe off-target effects. So we are currently looking into methods to also control this engagement by using, for example, bioorthogonal chemistry to cleave this engagement strategy. And just to show you if it has some promise, we are now looking into immunocytokine chemistry. This is a bioorthogonal reaction developed by the Tehran lab in 2017. And you introduce a specific immunocytokine molecule. It reacts with a strained alkyne, so a DPCO in this case, or a BCN. You have a cyclo-addition reaction, and you eliminate a citrulline molecule and a CO2 molecule, and you end up with this bicyclic conjugate. So to see if it works, we engineered our cells, we synthesized the building blocks, we loaded our cells with this cleavable linker, and we could see if we can then cleave this red color and change it into a blue dye in this case. And we were very happy to see indeed that we could have a dose-dependent change of our originally targeting molecule and switched it to something else. So this has great promise. We are now combining the nitrinium ion chemistry together with the synthetic CAR T cell therapies. We have recent ERC funding to do so, and our lab is now have a good manpower to unravel the reactivities of this nitrinium ion and combine it with synthetic CAR T therapy. And with that, I have to thank my former team and the current team that are very excited and very passionately working on all these fun playing with chemistry and modifying and targeting biology. And of course, the fundings and you for your kind attention.
Very much, Kim. Beautiful lecture with very complex molecules, both from chemistry and from biology. You see the applause and I see even hearts passing by. Lecture is open for discussion. Who has a question or a comment? I had a question, is that in this NOT enzyme, of course you need that to make this transformation from the N-linked acyl to the O-linked acyl. Is that always possible? So can you get the enzyme where you want it to be in the cell or on the cell?
Yes, so the enzyme, it's generally very low expressed in normal cells. So it's very highly expressed in metabolic cells, such as intestine and liver. But you can engineer this enzyme pretty easily. So you can introduce targeting molecules, or you can make fusion proteins. So or you can use viral inductions to engineer, in this case, our T cells, for example, to express it or overexpress it. And the nice thing of NOT is it's a human-derived enzyme, so it's not immunogenic. So we think it has a lot of benefits compared to other strategies that are currently on the market, which employs enzymes that are often derived from bacteria, for example.
Yeah, and does it need any cofactors or ATP or so?
So originally, if it has the acetylation, it has acetyl-CoA. But for our hydroxamic acid to nitrinium ion transformation, it doesn't need any cofactor. So that was very nice to see.
Exactly, that's very advantageous. Yeah.
Yeah.
Okay. Take your chance to ask Kim about her presentation, but I see that's not the case. So you have been very clear, Kim. Thank you once again for your contribution. And then we go to Professor Tati Fernandez from the University of Amsterdam. And we switch topic to homogeneous catalysis because we go to sulfur oxygen ligand promoted palladium catalyzed CH functionalization of arenes. So we stay into the arenes partly, but we are now going for CH activation. I see already your presentation, Tati.
Perfect. Thank you. Let me see. I will put it. Yeah. Thank you for the introduction and also for the invitation. It is a great pleasure for me to be able to share the chemistry that we do in my group with this community. And we mainly focus on the development of efficient synthetic methodologies. And for that, we use our strategy is to use the CH1 as a reactive functional group. And we combine that approach with the ligand design or catalyst design with mechanistic understanding, and also with new technologies like mechanochemistry to increase the efficiency of the reaction. So the main question is why we focus on the activation of the CH1 or CH functionalization. And I will highlight 3 reasons why we think this is really a relevant approach for the introduction of complexity in organic molecules. And the first feature is that if we compare CH functionalization with classical synthesis, the CH functionalization is just intrinsically more sustainable and more efficient. And here you have an example of the typical Suzuki coupling where you need to have pre-functionalized starting material with a halogen or a boronic acid. And these functionalities are not present in your target molecule, so they came out as a waste. In addition, these functionalities, they need to be introduced. If we look now at the bottom part of this slide and we use the CH1 as a reactive functional group, we not only reduce the waste that we form in this step, as we will form hydrogen or water, we also reduce the overall number of steps as these functionalities, they don't need to be introduced. So overall, we reduce the time consuming, the cost of the process, and the waste that we form. Another feature that I like about CH functionalization is that it allows the latest stage functionalization of more complex molecules. So already from a molecule that will have a biological activity, by using CH functionalization, we can just have many derivatives with probably different properties, and also allows them to increase the chemical space. And the last thing that I also enjoy about the CH functionalization is that it allows to have new retrosynthetic disconnections. And I always teach to the students, like if you want to introduce an electrophile, for example, in this aniline at the meta position, you cannot do that directly. So you need to do a functional group interconversion, and after you can introduce the electrophile. Nevertheless, if we think about the CH functionalization strategy and to using CH bonds as a reactive functional group, this retrosynthetic disconnection is now possible. So how we can do this activation of these CH bonds? There are many ways, electrochemically, photochemically, but the most common way and the way we actually use it in our group is to use a transition metal where the metal inserts into the CH bond to form a metal complex and then can be further functionalized. So at the moment, I told you the advantages of using the CH functionalization or CH activation approach, but there are of course some limitations, and the limitations are the low activity. We all know that CH bonds are unreactive. So in addition of using these transition metals to force the reaction to go, we generally use high temperatures, long reaction times, high catalyst loading, and this is an unsolved problem. And because of that, the yields are just moderate and we have a narrow substrate scope. Another problem is the selectivity, how we are going to control the selectivity in molecules that contain several CH bonds, like how we are going to control that the reaction occurs here in the hydrogen A or B or C. There are several approaches to overcome these limitations. We can, for example, use the intrinsic reactivity of a specific CH bond because maybe it's much more acidic, so that will be the reactive CH bond, but obviously that approach can only be used in certain type of molecules. And the most common approach that has been developed in the last 20 years has been to use directing loops. So the directing loop just coordinates to the transition metal and then deliver this metal in close proximity to this CH bond, facilitating the CH activation in a selective manner. And this approach is a very powerful approach and very successful, but has the limitation that you need to have in your molecule these directing loops. If the directing loop is not present, you need to introduce it and remove it. So the whole story about efficiency of CH activation is a little bit gone. So we thought that a way to go in this CH functionalization field was to do direct CH functionalizations of organic molecules without having these directing loops. And in that context, there were a couple of examples when we start our research reported by the group of you and Sanford, where they developed this or they discovered these pyridine-based ligands for the CH olefination of simple arenes. They use palladium as a catalyst in combination with this ligand. And as you can see here in this slide, they need to use an excess of the arene. So when you want to apply that chemistry to real organic molecules, this is just not, it will not fly. Wait a second. Now, sorry. So we thought that we think our idea was to develop a new type of catalyst that will promote this reaction and will allow us to use only one equivalent of the arene. So we came with this SO ligand, which was never used in metal catalysis, and there were only a couple of reports where they discussed the hemilabile properties of this SO ligand in palladium chemistry. So these ligands also are extremely easy to synthesize. So we test the reaction, we test these ligands in the reaction that was already reported, just to see if our ligand was more efficient to the ones that these pyridine-based ligands that they were described. So we use also an excess of the arene, the olefin, 5.1% of the catalyst, this oxidant, which is needed to regenerate the catalyst. And we were very happy to see, indeed, that already several of these ligands provide much higher yields than the reaction without ligand. The best ligands, they were the ones in this specific reaction that they were having an alkyl group at the alpha position and a phenyl group attached to the sulfur atom. We also saw that the reaction indeed was accelerated by the presence of the ligand. So with these conditions in hand, we evaluate, we try to understand the role of the ligand. So the first thing we did was to synthesize the sulfoxide and the sulfone, as probably we thought that maybe we have this active species as we have a strong oxidant in the media. Nevertheless, we observed much lower yields. So we rule out that the sulfoxide and the sulfone were the catalytically active species. We also kind of proved that the bidentate coordination was crucial as when we have only the thioether or the methyl ester, the reaction does not provide these higher yields that when the reaction with these SO ligands. And we also proved that the sulfur atom was key in this ligand. That's when we exchange by oxygen or nitrogen, the yields were much lower. So we, after that, did the screening of different substrates. And I'm not going to bother you with all these substrates, but I want to highlight 3 things here. The results in blue are the results in the presence of our ligand, and in black without. So in all cases, independently of the aesthetics and the electronics of our ion, we observe that in the presence of our ligand, the yields were much higher than without. About the site selectivity, the reaction was taking place mainly at the most electron-rich position of the ion, according to an electrophilic aromatic substitution. So for the whole presentation, you just need to think to understand the reactivity that this is kind of an electrophilic aromatic substitution where your palladium is your electrophile. And we also observe that the reaction, although we still have mixture of products, the ligand has some effect on the site selectivity. And I can highlight these 3 examples when we go to mainly para product without ligand, to ortho product in the case of this phenol or anisole, or here from 1-to-1 to 2-to-1 mixtures. So of course, this we can say, okay, it looks like this SO ligand activates the reaction, but still this, if we want to apply to complex molecules, we are still using this in excess, so will not be possible. But we were extremely happy actually to see that by modifying slightly the reaction conditions, they're using 10% of catalysts, we were able to do the functionalization of more complex molecules using only 1 equivalent of this arine. And you can now see the huge difference between the reactivity of the reaction without ligand and with ligand. So with these conditions in hand, we decide to move to now more complex molecules. And we thought to test our catalytic system in anilines, as anilines are widely present in bioelectrical compounds. And also because it was very interesting to see that there were very few reports reported at that moment that they could do the CH functionalization of a broad range of anilines. In most of the cases, what we observed was that the reaction, there were many papers when the aniline, for example, was unsubstituted or substituted with electron-donating groups. But there were only 2 examples where a broader subset scope was reported that also tolerates electron withdrawing groups. And the reason for that is because, again, this is kind of an electrophilic aromatic substitution. So when you have an electron withdrawing group attached to this aniline, you will need to force the reaction conditions to make it work. And the problem is that when we have here tertiary anilines with alkyl groups, these anilines are not stable under the standard conditions that we use in CH activation, meaning high temperature and the presence of an oxidant. So that was the reason why there were only these 2 reports on the CH functionalization of anilines bearing, for example, electron withdrawing groups. So we use the same conditions that I mentioned before with these simple arines, and as I mentioned before, these anilines were not stable. So we did not get the product, but we were extremely happy to see that just by modulating the reaction conditions, only at 40 degrees, we could do the para-selective olefination of a wide range of anilines. Also with electron-donating, electron-withdrawing substituents, primary, secondary, and tertiary anilines. And we expand this chemistry to indolinins, theta-hydroquinolines. We actually synthesize 10 different natural products that they all differ only in the structure of the olefin, so proving this latest stage functionalization that I told you before. And also other subsets were also tolerated, like thiophene or anisols. And we were also able to introduce other functionalities, triple bonds or arines. But moving on, and what I say at the really beginning of the introduction, that a good thing about CH functionalization is that in principle it will allow you to functionalize positions that they are unreactive. And so far, what I saw you before, what I presented here, is that the reaction is taking place at the para position. So at the most reactive position for electrophilic aromatic substitution. Nevertheless, it is possible actually to go directly to the meta position just using this norbornene, this mediator. Just to show you how the reaction works, in principle, you need to have the first CH activation that will take place at the ortho and para position. Let's put it like the para position. Then we will have the norbornene insertion here. And now this palladium is in very close proximity to this halogen, so we can have a second CH activation, further functionalization, and then termination to get the meta functionalized product, and we will recover also the norbornene. So in this context, there was one report in 2019 by the group of you where they used 50% of catalyst using these dual ligands, and they report the meta-relation of these anisols. But they mentioned as a limitation of this methodology that the reaction did not tolerate electron withdrawing groups attached to this anisole. In addition, in the supporting information, we found out that they also tried the reaction with anilines. And as I told you before, these anilines are not very stable, and when they found that when they are alkyl groups, they are not stable, or when you put an electron withdrawing group, a protective group like BOP, then again you are removing electrons, and then your aniline is not reactive enough. So because we reported the CH olefination of this kind of substrates, we decided to test if our catalytic system was also compatible with the presence of this norbornene, and to try to do this meta-olefination, meta-arylation reaction of this kind of substrates. And indeed, we were extremely happy to see that using our SO ligand, using the same conditions reported before by Jingwan Yu, we could obtain the desired product in very high yield. We also modified the reaction conditions, and we were able to reduce the catalyst loading to 5%, also the amount of norbornene, the silver, and the aryl iodide, and we get almost 100% NMR yield. Also some control experiments that the reaction without ligand almost does not work, also without norbornene, or the presence of silver in this case was needed as a halogen scavenger. So we did the substrate scope, and I want to highlight here 3 things. The reaction is extremely meta-selective. In this transformation, we did not observe any other ratio isomer. And we also have very high yields for all the substrates that we tested. With electron donating, they are all meta-substituted. I will come later for the ortho ones. We have electron donating substituents and also electron withdrawing substituents, which it was a previous limitation that it was mentioned in the previous report. We can also have tri-substituted anilines, and the reaction still works pretty well. We could also try different aryl iodides, again, electron donating and electron withdrawing substituents, they work quite well, and also different aryl bromides. In this case, you need to have here an electron withdrawing and coordinating group to facilitate oxidative addition. So then, as I mentioned, the substrate scope for meta-substituted anisols was very broad. But what happens when we have an ortho substituent and not a meta-substituent? In this case, the yields were very low, and that was expected because it is known in this Capellanian reaction that this substituent is needed to promote the norbornene exclusion. And if this substituent is not there, what it happens is, or you have a difunctionalization, or you start to have byproducts. Nevertheless, it was reported by the group of Dong in Chicago that a similar effect of the meta-substituent can be achieved when we have a bridgehead substituted norbornene. So we tried these bridgehead substituted norbornenes in our ortho-substituted anisols, and we were happy indeed to see that in these cases, we can have very high yield of the meta-related product, and still the site selectivity is extremely high. We test other substrates here, and again, very general for electron donating and withdrawing substituents. Then we move our attention to see if we could have the monoarylation of anisols, which is still a very difficult task in this CH activation field. So we test different bridgehead substituted norbornene, and we found out actually that this one, this N10, gives us a very high monoselectivity and a very high yield, and this reaction seems to be general for different substituted aryl ethers. And considering that we were able to introduce only one aryl group, we thought that then maybe we have the possibility to introduce two different aryl groups. So indeed, it was possible, and we just play a little bit with the reaction conditions. In the first step, we just use this bridgehead substituted norbornene to be sure that we do the monoarylation. Then we filter through a part of sea light, and we add the second batch of reagents, and now this norbornene, and we were able to introduce this second aryl group. And although the yields here are moderate, I would say that it is difficult to imagine a direct process where you take anisole and you can functionalize with two different aryl groups at the meta position of this substance. So we also did some mechanistic investigations here to understand a little bit what was going on in our catalytic cycle. So we were very lucky that we were able actually to isolate these complexes just using this anisole and one equivalent of palladium, ligand, and norbornene. And this reaction, that means CH activation and norbornene insertion, could even be done at room temperature, indicating that probably these steps are not rate limiting. Also, by adding a base, we could isolate the complex coming from the second CH activation. And we tested these both complexes in their reaction, and we proved that they were catalytically active, and they gave the arylated product in more or less similar yield. Furthermore, we took this complex before the second CH activation, and we put the deuterated hexafluoroisopropanol under the same conditions, and we observed the introduction of deuterium, indicating that at least in an isolated manner, this step is reversible. So our picture at that moment, and we have much more information here, is that we have the CH activation is taking place at the para or ortho or para position, then norbornene insertion, and we know these steps are all reversible because actually when we follow the reaction, we can observe the formation of this complex that after a while it comes back to the starting anisole. Then we have the second CH activation, which in principle is also reversible, and then follow it by the oxidative addition and reductive elimination. And our studies indicate that probably oxidative addition is rate limiting in this case. So what about the anilines that I mentioned before? There was this limitation that at the moment nobody could use these anilines in this meta-functionalization. So we test these anilines now with our catalytic system, but we only observe very low yield. We use this 10% of palladium, of the ligand, of the norbornene, with in the presence of silver. In this case, we use chloroform as solvent. In the previous case was hexafluoroisopropanol dichloromethane. And yeah, here we have very low yield. And as I mentioned before, we think that the problem is the oxidative addition. That is difficult. So we decide to cheat a little bit here and to use this aryl iodide, which is more prone for the oxidative addition to form from palladium 2 to palladium 4. So under using this aryl iodide, we obtain this 48% yield, which could be further improved by modifying the norbornene. In this case, we use this amide norbornene. Then we did the substrate scope, and again, very general. Extremely meta-selective, we did not observe any other regioisomer, and very high yields in all these cases. In red is just the reaction without a ligand, and it's not nothing to do with this normally. So yeah, very high yields for electron donating and withdrawing substituents. And then we also test now these Boc protected anilines, and the reaction also works. In this case, we need to use this ligand L4 here, which is it's also reactive with this phenolic structure, and this norbornene. We know when to use different norbornenes. I would say when this hydrogen, this meta hydrogen here is more acidic, the ester norbornene works well. When it's less acidic, like when you have these tertiary anilines, we need the amide norbornene, because this amide norbornene promotes the second CH activation, as we will see later on, just to explain a little bit the structure. So we also test here different orthosubstituted anilines. Again, in this case, we need to use the bridgehead substituted norbornenes. And we are able to do this meta-arylation, very highly selective. In these cases, maybe the exception is this one because we have an electron donating group, but in most of the cases it's very high selective. And we could also introduce different aryl iodides, but in all cases with this ester group or with this electron withdrawing groups and coordinating groups, that they help this oxidative addition to take place. So in principle, and that will say, okay, it's relatively general because we can afterwards remove the ester, but we were really eager to have a much broad scope of aryl iodides. And because we were having a better understanding of the reaction, so we decided to again study the reaction with this aryl iodide. In this case, we found out that using this SO ligand, the reaction gave slightly higher yields. And this is because this SO ligand, we did some calculations, and the palladium complex is a little bit more electron rich than when we use this SO ligand, so oxidative addition is more facile. And also, we use a different norbornene, and our idea here was to have electron withdrawing groups far away from this double bond to again make your palladium a little bit more electron rich. And under these conditions, actually, we were able to obtain the meta-related product in relatively good yield. And the scope was relatively broad. We just test only these 4 substrates because we did a lot of substrate scope before. So as I mentioned, we also studied the complexes, and again, we could isolate also from anilines these complexes. And what we studied was the effect of the substituent here in the second CH activation, as well as the influence of the norbornene that we use. And we observed the following. In this, we observed that when we have no substituent here, and we use the ester norbornene here, no deuterium incorporation was observed. So no second CH activation was occurring. Nevertheless, when we move from this unsubstituted aniline, but instead of the ester, we have the aniline, we observe that we observe the incorporation of deuterium, meaning that this amide norbornene is helping in this second CH activation. This is why in some cases we need this amide norbornene. Furthermore, we did the experiment using this metal complex, using the metal complex that have a methyl group here. In this case, when we have an ester norbornene, we observe that we could also introduce deuterium at this meta position, meaning that this methyl group, in addition to promote the norbornene extrusion, it also promotes the second CH activation step. So overall, I hope I convinced you that we have a very powerful catalytic system for the functionalization of diverse type of substrates, and moreover, we could control also the site selectivity where the reaction takes place, if it's at the para or at the meta position. Then I would like to thank the people that did the work in the lab, which are this one. This is an old picture, and mainly this is the work that I presented today is done by Karanat, Moenian, Bibi, and Ketsuan. And I will also take a little bit of time to promote the first Spanish Organic Chemistry Symposium that it will take place in the ICIQ in November 17 to 19. And the abstract submission deadline is 30 of September, so you should hurry up if you want to present a poster or a presentation. And the early bird registration is on October 8th. And thank you for your kind attention.
Thanks very much, Tati. Symposium highly recommended. And thanks for your insightful lecture. And counterintuitive for many students joining currently the presentation, I think, being used to electrophilic aromatic substitution. Okay, question. The lecture is open for discussion. Who can I give the floor for a question or a comment in the chat box? Maybe I can start because I see you switching every now and then in terms of oxidation reagent. Is that just because you get higher or lower yield, or what's in that reagent? Because sometimes you use a perester and sometimes you use silver acetate.
Yes. Okay. In general, if we have an anhydride iodide, you need the silver because you will form palladium iodide, and that is unreactive. You need the acetates in your molecule to remove the proton. Let's put it like that. So you need the silver to abstract the iodide. But silver, in some cases, like this one, for example, I don't know if you still see the presentation here. Silver, we now submit a paper where we prove that silver is also needed in this case to promote the oxidative addition. As I mentioned, the oxidative addition is rate limiting, and we need this silver. So it has several roles in this case. But if, in principle, if we don't have any iodides, we can use other oxidants, even oxygen. In the olefination reaction, we can just use oxygen, or air also works.
Okay. Okay, great. And if I made the use of aryl diazonium salts in these cross-coupling reactions, would that also be possible?
Sorry, again?
The use of aryl diazonium salts in these cross-coupling reactions, would that also be possible? Then you don't have an iodide, at least. Or
Yes,
at
yes,
least.
I think, yeah, I think so. Yes, I don't see why not. There are, I think, few papers that they use it.
Okay.
Yeah.
Okay. Thank you. Yeah, I see a question coming in from Leonard Estelle. By using your ligand, do you avoid the formation of nanoparticles from palladium acetate? Is the question.
The question is maybe if I stabilize the palladium. No, I think we still form. I mean, we observe the formation of palladium black still, and therefore I think also nanoparticles. So no, I don't think. I think it's just that we really accelerate the reaction. But palladium nanoparticles or palladium black is still being formed.
Okay. I see no more questions coming in, so let's thank Tati once again for her contribution.
Thank you very much.
And we stay in Amsterdam, but we go to the other university, Professor Elko Ruiter, Amsterdam Free University. Cascade reactions for the total synthesis of complex indole alkaloids, serendipity versus design. Elko.
Thank you very much, Adri, for the kind invitation, and also, of course, for the invitation to speak among this wonderful lineup of speakers on very, very diverse topics. And if everybody can see my presentation well?
I do see your presentation, not yet in full presentation mode.
This happens sometime.
Mm-hmm.
Let me just try and... Better now?
Yes, now I see it in full presentation mode. Yeah.
Yeah, so indeed, I'd like to speak today about the work that we have done in our lab in the field of synthesis of these complex indole alkaloids using cascade reactions, which is one of the main topics in our group. These indole alkaloids, most of which are monoterpene indole alkaloids to be precise, are among the largest and most diverse class of natural products known to man. And ever since I chose a chemistry career, I've always been fascinated by, let's say, the molecular aesthetics of these compounds. They contain many different ring systems, fused, bridged, many different stereocenters. And I've been wondering from very early on, how does nature make these molecules and how could we make them? And this has been a challenge, of course, for many people in the field. Natural product synthesis has been a major topic in organic chemistry for many years now. And I think historically its role played a large role in understanding how nature makes these compounds, understanding biosynthesis, confirming the structure that was assigned to these natural products, especially before the advent of modern spectroscopy techniques, but also as a showcase of what organic chemistry at that particular stage in history could do. In a broader context, though, more pragmatically, many of these natural products, especially these indole alkaloids, have considerable and interesting bioactivities that might be useful for medicinal applications. But not by necessity is the natural product itself the most active compound towards the target we would be interested in. So synthesis, by definition, can provide access to many interesting derivatives of these compounds, but also provide access to useful quantities of what I would like to call rare natural products, because many of them have been isolated in rather minute amounts from rare sources of not only plants, but also marine organisms, for example, that may not even be found back on another expedition. But I would say in a broader context, natural products provide an ideal testing ground for the development of new synthetic methods. The diversity and complexity of the scaffold structures of these molecules is immense, and techniques that allow us to construct molecules like this obviously have a much broader field of application than only these molecules alone, and therefore might have a broader impact on synthesis in general. Secondly, they contain typically a large number of functional groups and thereby inherently test the functional group compatibility of the synthetic methods that we develop. And because there are so many stereocenters, we also are forced to think about all the stereochemical consequences of the molecules, not only in establishing these stereocenters themselves, but also in the consequences of the 3D structure of the molecule for the reactivity of specific functional groups. Specifically quaternary stereocenters, so that is stereogenic carbon atoms attached to 4 other carbon atoms, may be very hard to produce indeed. And also, because of these complex structures, many of the disconnections that we would like to make may not be accessible through current methodologies, and developing new methodologies to do so can provide access to, like I said, a larger number of molecules even. So our take in the field is to use cascade reactions. And what essentially is a cascade reaction? I've pasted this picture of a waterfall. And essentially what happens in a cascade reaction is an activation of a starting material by some sort of activation method to give a reactive intermediate that can be cationic, anionic, radical, or metal bound, that proceeds through a series of well-defined, thermodynamically feasible elementary chemical reaction steps, with each of the intermediates undergoing a defined subsequent elementary step. And this makes sense from an efficiency perspective, because what do we want to achieve with synthesis? This is essentially making and sometimes breaking covalent bonds. If we can combine several of these processes in a single reaction, that inherently increases the overall efficiency of multi-step synthesis. Now, coming back to the structures of these monoterpene indole alkaloids, and today I've shown 4 of the key players in today's talk. They all share some commonalities, and this pyrroloindoline moiety, highlighted in blue, is one of the key hallmarks of this large family of natural products. But as you can see, the connectivity of other atoms and other fused or bridged rings may differ quite significantly. So if we develop the right kind of chemical tools, we might be able to access multiple of these structures by using an overarching strategy to achieve them. And there are essentially 2 different ways to make these molecules, and I've made them into 3 here. And the first, obviously, is to start from some kind of indole-derived starting material, most commonly a tryptamine derivative. This is a good idea in principle, because this is in some ways biogenetic, because all of these compounds are biogenetically derived from tryptamine. So from a synthetic perspective, it would make sense to also start from tryptamine derivatives. This is one of the examples that we've shown earlier, to use the unique reactivity of isocyanides to construct these pyrroloindoline intermediates, which we were then able to convert further to this molecule spidofractanine. On the other hand, we can also choose to build up that rather reactive indole ring only at a later stage in the synthesis, by a late-stage Fischer-Indole synthesis, for example. And this has been done by many people in the past. Or alternatively, we could construct the indole motif via a reductive cyclocondensation of nitroarenes. So, and I will basically focus today on the first and the third strategy. But each one of these has its own pros and cons. And evidently, using tryptamines has the advantage of being atomo-economic, in the sense that we have readily available starting materials that already contain a large number of the atoms present in the final target. But on the other hand, you're really reliant on the functionalization, the appropriate functionalization of that precursor. And because both indoles and indolines are rather reactive and nucleophilic species, they often require protection over the course of the synthesis. Late-stage indole formation through a Fischer-Indole synthesis also has some potential benefits, because it adds in a single step a lot of the functionality that you need, so the entire indole system from a ketone precursor. But there's quite often considerable regioselectivity issues with this approach, because cyclization may take place in 2 different potential directions, leading to 2 different regioisomers, of which only one is desired. And as it turns out, this is quite difficult to predict. So the final strategy, the reductive cyclocondensation of these nitroaryl substituted ketones, prevents that those regioselectivity issues, but actually it sort of replaces the problem with another problem. How do we site selectively introduce that nitrophenyl group here? And this will be the topic of the second part of my talk. So the first part will focus on the first strategy, using tryptamine derivatives to construct the scaffolds of these types of molecules. And this all started with a bit of a serendipitous discovery. So several years ago now, we were working together with a biocatalysis group in Manchester led by Nick Turner, in which we investigated the biocatalytic reduction of these dihydro-beta-carbolines that we can synthesize very efficiently through the Bischel-Napieralski cyclization of these tryptamine-derived amines. And the Manchester group had several different amine reductases that were able to, with very high enantiomer selectivity, reduce these cyclic amines to the corresponding secondary amines, and we could subsequently further modify these to these somewhat simpler indole alkaloids. And Ras van Chockel was working on this project at the time, made about 20 examples of these dihydro-beta-carbolines, and all of them proceeded in very good yield, except for one. When we use this specific tryptamine-derived amide in the Bishop-Napierowski cyclization, we did not observe even a trace of the expected dihydro-beta-carboline product. Instead, we isolated a different, rather apolar compound, which we call compound X at the time, because initially we could not really figure out the structure. But we found that the reaction was very selective and very efficient. In about 95% yield, we observed a single product from this reaction, but curiously, it did not contain any aliphatic carbon atoms. And MS analysis also showed that we lost a CH2NH fragment, so likely this part of the molecule. So this seems very strange. We lose the middle part of the molecule. So fortunately, we were obtained some crystals that were just enough quality for X-ray analysis, and our collaborator Chris van der Velde from the University of Antwerp was finally able to establish that the reaction product was this phenyl-substituted carbazole. And at first, we did not understand this very well, so we ended up shelving this project for several years, actually, before returning to it. But you could argue that our carbazoles in their own right have some merit, and because these are some naturally occurring and synthetic carbazole derivatives that have shown biological activity, but they are not quite as diverse, of course, as the indole alkaloids that I've shown you before. So we shelved the project for quite a while until Matteo Valtracco picked it up a couple of years ago, and we still did not understand the mechanism very well at the time. So Matteo just started to systematically investigate the substitution of the reactant. And so what Matteo did is just introduce methyl groups at several positions of the starting material, just to see where they would end up in the product. So if we put the methyl group next to the carbonyl, it ends up meta with respect to the phenyl. That makes sense because they also have a 1,3 relationship here. And not surprisingly, if you move it to the beta position of the acyl residue, it ends up right next to the phenyl group. So what then if we put it on the same carbon? Well, so in that case, we formed a complex mixture from which we could not really isolate any single product. But the real breakthrough came when Matteo made the 2-methyl substituted indole derivative. And quite to our surprise, that methyl group ended up at this position here in very high yield. And this to us could only mean one thing, that in one way or the other, the indole fragment in this molecule must be broken or deconstructed during the cascade reaction. And this then led us to formulate the following mechanism for this reaction, which starts off with the regular mechanism of the Bishop-Napierowski reaction. The secondary amide is converted by the action of a phosphoryl chloride to a nitrilium ion, which is then attacked by the nucleophilic indole at the C3 position to give this spiro intermediate. So typically in a normal Bishop-Napierowski reaction, you would get a so-called Blanchet rearrangement, so the 1,2 cationic shift, to give this tertiary benzylic carbocation, which is rapidly deprotonated to return the indole-containing product of the regular Bishop-Napierowski reaction. So something must be different in this case. And considering that we have quite acidic reaction medium, we argue that perhaps this imine could undergo tautomerization to the corresponding enamine, which is then conjugated with the styrol moiety. So the resulting aminodiene is then perfectly positioned to undergo a vinylogous Mannich reaction with the imidium ion of the indolenine. So this would lead to this tetracyclic intermediate, which looks much more interesting, in fact, than the starting material or the product of the reaction. And at this stage, we think that after migration of the proton, the amino group, or the aromatic amino group, undergoes a beta elimination to give this highly conjugated diene. And then we have both a free amine and an iminium ion. So this can then be in equilibrium with the corresponding aminal, but also open up towards the other direction again to form something that already looks quite a lot like the product. And the only thing that we really need to do at this stage is cleave this carbon-carbon sigma bond. So if any nucleophile would attack at this CH2 group here, it could expel the phenyl substituted carbazole as the product. Since the only reasonable nucleophile in the reaction mixture is chloride, we believe this is actually chloride in this case. Of course, this mechanism raised some questions. So first of all, we would like to further confirm this by taking a look at, see if we can label this carbonyl group, for example, and see if it indeed ends up at this position as a further support for this mechanism. So this does not seem trivial because we would need a carbon-13 label at this position of the starting material. But fortunately, we found this method in the literature, the palladium catalyzed carboxylation of cinnamyl alcohol using palladium acetate and xanthos, followed by the in situ trapping of the resulting carboxylic acid with the tryptamine to essentially make that carbon-13 labeled precursor in a single step. And indeed, when we subjected this to the reaction conditions, we obtained the product in high yields with the carbon-13 label at exactly the right position. So then, of course, we wanted to see what the generality of this reaction was. And in fact, we found that we can substitute both the indole fragments and either on the benzenoid ring or the N1 position, or on the sterile acetic acid moiety. We found that we can vary all three quite considerably. We could also replace the arene by heteroarenes or carboxylic esters, of which this particular one at the bottom is actually a natural product, showing that the reaction is actually quite broad. But still, we were quite puzzled by the reaction mechanism, and to see if we can get further support for this proposed reaction mechanism, we wanted to see if we could divert the course of the reaction at some stages. And especially this tetracyclic intermediate would be a nice point to divert the course of the reaction. So the question we asked ourselves is, what if we would, instead of the hydrogen atom here at the ring junction, have something that is a better leaving group than the protonated amine, for example, a bromide. So we went and synthesized this precursor that has a bromide at the 2 position, and the reaction would follow, in this case, the mechanism as shown before, up to this point, where the bromide would be the better leaving group compared to the aromatic amine. This would then lead to this intermediate, but as it turns out, the reaction does not stop there because the aromatization towards the carbazole provides a too high driving force. And what we observed is that, in fact, chloride does attack as a nucleophile here and gives us these chloroethyl aniline derivatives or aminocarbazole derivatives. Also here we were able to show several examples, and all of them provide that rather reactive looking chloroethylamide, a chloroethylamine derivative in quite good yields. So I must say that we also have some of the corresponding bromide because the mixture contains both chloride and bromide atoms. So we could very clearly see both the chloride and the bromide patterns in MS analysis. As a further support for the mechanism, we wanted to investigate what would happen if we could somehow further divert the course of the reaction at this stage. If indeed we would have a nucleophilic substitution of chloride, in this case, on this CH2 atom, can we divert the course of the reaction by replacing it with something that does not undergo SN2 substitution? So we decided to replace the ethylene linker by a phenylene linker. So we synthesized this starting material instead, and the reaction indeed follows the same course as before, but up to the point where we have this iminium ion, which can form this butterfly-shaped MNL, which can open up again, but this is a symmetric species, and these two iminium ions are in fact the same. So there's no possibility here for the amino phenyl group to be attacked by chloride and undergo SN2 substitution, but we do have a rather electron-rich arene and a positively charged system. So what happened in this case is that the orthoamino phenyl group undergoes a 1,2 cationic shift to give this amino phenyl substituted carbazole instead. And actually, in this case, it does not matter which of the two undergoes that cyclization, undergoes that reaction, because they are both identical. So, but finally, so all of the products that we had isolated up to this point are still flat and aromatic and not these nice 3D structures that we appreciate in the indole alkaloids, although the reaction was proposed to involve these types of tetracyclic indoline derivatives. So we were looking for ways to somehow stabilize or interrupt the reaction mechanism at the bottom right stage here. So this led us to another question: can we divert the course of the reaction in another way, or more precisely interrupt it at this stage? And of course, the beta elimination requires a proton at this position. So what if we would have two different carbon substituents at this position? So finally, this brought us to start from this diester, so a malonate derivative, if you will. And indeed, if we expose this to the reaction conditions, the reaction is interrupted at the stage of the Vineligis-Manich reaction. So I will not get into the details of how messy a reaction this is to make the starting material, but looking at the structure of this tetracyclic product, we thought this would be a good starting point to complete the total synthesis of this mu-opioid receptor ligand aquamycin. And we thought if we could do a selective 1,2 reduction of this alpha-beta unsaturated imine, then after alkylation and decarboxylation, we could get this iodo-vinyl derivative that can undergo a intramolecular Heck-type reaction to arrive at aquamycin. Unfortunately, we were not able to selectively reduce the imine in the presence of the alkene. In one way or the other, either nothing happened or the alkene was reduced in a 1,4 reduction. So we decided to sort of protect that alkene by conjugate addition of thiophenol, after which we could readily reduce that imine again, alkylate, and then treatment with MCPPA followed by heating eliminated the thioether again to reform the alkene at that position. So then careful treatment with potassium hydroxide selectively hydrolyzes one of the two ester moieties, followed by decarboxylation and alkene migration to arrive at the final intermediates, which can indeed undergo the final Heck cyclization in very good yield, completing the total synthesis of aquamycin. So this is sort of the end of the first part of my talk, because we discussed that the indole dearomatization is one of the potential routes, and I hope I've been able to show you in the previous example that we can actually couple the formation of this 5-membered ring to the formation of another ring system that is required for the synthesis of these compounds. But another approach that I mentioned is the reductive cyclocondensation of these nitrophenyl substituents, either with the pyrrolidine ring already formed or not, which I already mentioned has the challenge of installing this nitrophenyl moiety in exactly the right position. So this will be the topic of the second part of my talk, in which we will discuss the synthesis of, amongst others, these two natural products, which we both thought would be able to derive from this pentacyclic precursor. And this precursor we thought we would be able to conveniently access from this nitrophenyl substituted alpha-beta unsaturated ketone. But as I mentioned, installing that nitrophenyl moiety at this position is not easy and often requires a multi-step synthesis. So, inspired by some literature precedents, we thought we would be able to achieve this by a nitroaryl transfer cascade reaction from these orthonitrobenzene sulfonyl amides, which would result in a formal 1H shift of this orthonitrophenyl group. And of course, this is not a direct process, and I will go into the mechanism in a bit. But this precursor we thought we would be able to conveniently access from cyclohexane dione. So this will be the key reaction that we will be discussing today. But first we will start at how we arrived at this precursor that already bears the quaternary stereocenter that we will need to induce all of the other stereocenters. So starting from cyclohexane dione, we first protect as an enol ether, after which we can readily generate an enolate at this position, install the allyl ester moiety, and after which we can deprotonate again and do a conjugate addition to acrylonitrile to arrive at this racemic intermediate. So also the racemic intermediate has a quaternary carbon stereocenter, and we were able to desymmetrize this fragment using palladium catalysis. So this is a decarboxylative allylic alkylation reaction. So what essentially happens is that the palladium catalyst with a chiral ligand inserts in the allyl ester moiety, undergoes decarboxylation to form this palladium enolate, which then reinstalls the allyl fragments on the alpha position of the ketone, which proceeds with high enantiomer selectivity because of the chiral ligand on the palladium center. So we found this to be a very robust reaction, and we could reduce the palladium loading down to 2%, perform this reaction in larger than 15 gram scale to obtain this stereo-defined precursor. So we then reduced both the ketone and the nitrile, and in our hands this worked best if we subsequently treat it with dibol and then with lithium aluminum hydride, followed by the addition of nosyl chloride to install the sulfonamide here, and an aqueous workup hydrolyzing the enol ether and eliminating water to arrive at the alpha-beta unsaturated ketone via a Stork Danheiser transposition. So we've now conveniently accessed this cascade cyclization precursor. So what is then happening in this reaction? So in this case, the reaction proceeds via an anionic intermediate. So clearly we can deprotonate, in this case with cesium carbonate as the base, the highly acidic sulfonamide, which can then undergo an intramolecular conjugate addition to the alpha-beta unsaturated ketone, resulting in this enolate, which can in turn attack the very electron-poor arene via this Meisenheimer type intermediate. Now we would call this a true Smiles rearrangement, and via this sulfinamide, we would get to this deprotonated amide, and after a proton shift to the enolate here, which can also undergo an E1CB elimination again. And we believe that in the reaction mixture, we have a dynamic equilibrium between, well, these two species, which is why we decided to trap the primary amide with the chloroacetyl chloride, which would be the next step in the synthesis anyway. So even though the isolated yield of this reaction is not very high, it proceeds through many different intermediates. This is really the only isolable product, and we think the rest of the material is probably some oligomers that are not really well soluble in the reaction anymore, which makes this product actually rather conveniently isolated. So there's a bit of hand waving in this mechanism. I will come back to that a bit later. But of course, we also wanted to show, does this method have any generality? Can we add additional substituents on that nitrobenzene sulfonamide? And indeed, we can. We can install some halogens or a CF3 group at this position, and even a methoxy group at the meta position compared to the nitro. But when we move the methoxy to the ortho position of the nitro substituent, the reaction does not work anymore. And this is actually quite readily explained by the fact that we require stabilization of an anionic intermediate from the nitro group. And if we have the methoxy next to it, there's already considerable delocalization of the lone pairs on the oxygen atom of the methoxy group towards the nitro group, making it less available for the stabilization of an anionic intermediate. Replacing the nitro with the nitriles, unfortunately, does not work. So this then sets the stage to complete the total synthesis of various different skeletons of different classes of indole alkaloids, starting in fact with another cascade type reaction. If we take the cascade product and treat it with iron and acetic acid, we reduce the nitro group to the corresponding amine, resulting in condensation onto the ketone, generating a very electrophilic intermediate at this position, which is then attacked by the secondary amides to form this tetracyclic indole derivative. And conveniently, this is a highly crystalline compound, which allowed us to increase the EE of 91% to over 99, and also measure the crystal structure, showing the cis fusion of the two rings in this tetracyclic molecule. So at this stage, we can also install the 5-membered ring by activating this chloride, leading to the common intermediate that I mentioned before, containing 5 of the rings of the target molecules. So the first molecule we decided to target was limospermidine, and this really required the complete reduction of this molecule using lithium aluminum hydride, which gave this intermediate in quantitative yield, or in 89% over 2 steps, because this common intermediate here is not a very bench-stable intermediate. And then ozonolysis under acidic conditions to protect the 2 amine functionalities, followed by a workup with sodium borohydride, immediately afforded minus limospermidine. So similarly, we could convert this common intermediate to the corresponding azaenolate with LDA, followed by treatment with Mendes reagent to install the methyl ester here. After that, we Bock protected the indoline, which proved to be necessary in this case, after which we can again, by ozonolysis, cleave the terminal alkene to generate the primary alcohol after reduction with sodium borohydride and substitution of the primary alcohol by an iodide. This primary iodide was another branching point in our synthesis, because if we now add samarium iodide, we can get an iodine atom abstraction to generate the primary alkyl radical that adds to this position to install the bridge in this way, in a bicyclo 2,2,2 octane, to give, after Bock deprotection, copsinylam, which is also a natural product, and after a 2-step removal of the carbonyl oxygen, we also completed the synthesis of minus copsinin. Conversely, if at this stage, at the stage of the primary iodide, we decide to remove the Bock group, this results in a rather nucleophilic enamine that displaces the primary iodide by SN2 substitution, providing this bicyclo 3,2,1 octane framework common to the copsifolines. After reduction of the resulting amine with sodium borohydride. And after 2 additional steps, removing the carbonyl oxygen, we arrive at the tetrahydrocopsifolene B. So all of these materials, again, were accessed from this one common intermediate. So still, the mechanism kept with us for a bit, because we wondered, this intermediate here, this is an amide ion. It's a deprotonated amine, and it seemed an unreasonably high energy intermediate. So this led us to study the mechanism in some more detail using DFT calculations, which were performed by Thomas Hansen, assistant professor in the group. This soon proved to be much more complex than we had initially anticipated, because even the starting material can adopt to different conformations which are distinct and have their own distinct reactivity. Moreover, over the course of the reactions, we generate new stereocenters, and intermediates might be cis or transfused. So without going through the complete mechanism, there is only a few pathways that are even feasible that led to the product. But one observation that we made is that this Meissenheimer intermediate is not at all a reaction intermediate in this case. And that reaction does not involve an amide anion. Instead, we break this carbon-carbon bond to break open the cyclohexane ring to give this enolate, which only reacts further to the final product after protonation of the cyclic imine to reform the cyclohexane ring. Do I have a little bit more time, Adri? Or should I wrap up?
No, you should about wrap up, yes.
Yeah. So this then led to this revised mechanism, which indeed involves the ring opening. And the final question that we asked for, can we then also replace that substituent that we had here to make a fused product in the cascade reaction to a spiral product, a bridge product, or an alternatively fused product? And to cut the long story short, we computed the initial stages of the reaction, and we found that certainly these two were not feasible, but the spiral was just about. The only thing that we really had to tackle was reduce the second transition state, this TS2, for a little bit. And we chose to do so via the introduction of an additional nitro group on the aromatic ring, lowering the transition state for the second step. And in this case, because we have additional stabilization, the reaction does proceed via a Meissenheimer type intermediate. Unfortunately, in this case, this led to this more linear product because the corresponding enolate here is not nucleophilic enough to undergo the manic cyclization to the expected spiral product. So, and now I really am going to wrap up. I hope I've convinced you all that these serendipitous findings can be a really fertile ground for new reaction discovery with applications far beyond the initial findings. And cascade reactions are indeed excellent tools for natural product synthesis owing to their excellent atom and step efficiency and high chemo and regio selectivity. And also advanced mechanistic insights and resulting better understanding of reactivity can result from both thoughtful control experiments and DFT calculations, or better still, a combination thereof. So, of course, I want to thank all the group members involved and the collaborators and the funders, of course, and you for your kind attention.
Thank you, Eelco, for a beautiful lecture in natural product synthesis. And also you deserve the applause and the hearts even. Good questions, comments to be put in the chat box. I myself was wondering, but it's maybe difficult to get it back, that in the first part of your lecture you basically interrupt this complex rearrangement to the carboxyls, and at some point I wondered, could you even interrupt it at an earlier stage? But I cannot explain exactly what I mean.
Let's see. So I think, so you mean at the stage of the spiral cyclization, for example?
Exactly, yeah.
Yeah, so that is indeed very difficult because the barrier for the Planchet rearrangement to take place for the ring expansion to give the beta carbonyls is not really very high. So you really have to divert the reaction at this stage in order to prevent the Planchet rearrangement from taking place.
And another question I had, but for that it was a bit too complex for me, that sometimes I thought, yeah, these are all, let's say cationic, anionic reactions. Are there also pericyclic reactions happening in these kind of rearrangement, that you see a Cope or a Claisen type rearrangement?
Yeah, so this is indeed a very interesting and very promising reaction. And in fact, we do have one project ongoing where we also have a Claisen rearrangement involved. So stay tuned, I would say. But others have used pericyclic reactions as well. And in fact, you could also write up alternatives for some of the mechanisms that I've proposed here that are more pericyclic in nature.
Mm-hmm. Okay. Okay, thank you. I don't see questions coming up in the chat box. Yeah, then you missed the chance, of course, but apparently it's very clear. Thank you very much, Eelco. And then we go to the final lecture in this series, and that's provided by Professor Hans Zuilhoff from Wageningen University. And he will lecture on the synthesis of chiral sequence-controlled oligomers using suffix and suffix click chemistries. Han, the floor is yours.
Well, thank you very much, Ali. So let me open here.
I do see your presentation.
Good.
But not yet in full. Yeah, there it is. Yeah.
There you are?
Yeah.
Okay, good. Well, first of all, thank you, Adri, and other people involved in the organization of today. I'm very happy to have the chance to wrap up this excellent series of lectures. And I decided to give a somewhat broader view on both intrinsically chiral click chemistry and multimodal click chemistry. And this will end up with the sequence-controlled oligomers, sequence-defined oligomers and sequence-controlled polymers that the title referred to. Before I go there, I first have to thank the people involved in the work. That is my group in the Netherlands, in Wageningen, the guys you see over here. And there's also two groups, two of my labs in China involved, namely my lab in Tianjin and in Jiaxing. And I have to thank them all. And as you can see, the bunch here in Tianjin, they really bring me to a higher level and also spur enthusiasm complementary to the other groups. So very happy there. So what are click reactions? Yeah? We're going to speak about these. So if you just take a simple-minded view, then you'd say, okay, these are type of ideal reactions. You take two mild reagents, you mix them at room temperature, basically, then you go away, drink some coffee or tea, and then you're done. And this should be a reaction that gives a high yield, no toxic waste, and provide an easy purification. So basically, if you have some simple, well-chosen building blocks, you put them together, and actually then you get a complex structure. But of course, if you remind what the Nobel Prize in 2022 was given for, you actually see that the definition is a bit more precise. Yet, this is the definition that people typically take, namely, coming from Sharpless or derived from Sharpless lecture in 2001, he defined click reactions as reactions that are modular, wide in scope, give very high yields, generate only inoffensive byproducts. Then basically very many people think, and that's all, yeah? But in fact, what the paper described was this. These reactions should also be stereospecific and either use no solvent or only a benign solvent, such as water. Yeah? So what I'm going to do in this lecture is focus on the modularity, on the stereospecificity, and on the use of no solvent in these type of reactions. So first, regarding the modularity, can we use modularity in click chemistry of polymers? Suppose you have a building block, yeah, here the blue block, and then you couple that via some click reaction to another block. You have a dye block, and of course, we being clever, we can just repeat. So in this way, we can make polymers. And of course, if you take a somewhat different scaffold, here with three sides, you can make more complex structures, etc. Then, of course, what people have done in polymer synthesis is you first make in this fashion a polymer, and then using first the, in this case, horizontal sides of nature 1 to make the polymer, then use an orthogonal reaction to construct something that can be added onto the polymer. And finally, typically another reaction is needed to get the functionality of interest. So when we entered this field, what was done was, let's say, something like this. Many people have done this, yeah? Orthogonal a clicking of different moieties. So it would be stepwise building. But what had not been done is, can we actually tune a click reaction in such a fashion that it becomes sequence sensitive? That is, that we can first make the polymer and then using the same reaction, do the derivatization. Of course, you don't want to end up with this, where both the polymer formation here horizontally and the functionalization here vertically happen at the same time, because basically then you get a mess, or as other people like to define it, a network. When we started this field, we thought, okay, what type of reactions would be useful to do this? And here we were drawn to the sulfur-fluorine exchange reaction. Now, the reason is this paper from Sharpless in 2017. And this suffix chemistry, and here you see an example, couples the sulfur to a nucleophilic entity. Here it's the oxygen of a silicon-protected phenol. What it does is the oxygen attacks onto the sulfur, kicks out the fluoride, and then you form basically a sulfur-oxygen bond. And if you do this in a difunctionalized species with a difunctional phenol, you get a polymer. Now, why is this so interesting to study with the polymer? Then you can really investigate the yield of this reaction in much detail. And for this reaction, the reported yield was 99.9999987%. And that you can just derive from the molecular mass and distribution of this compound. And I thought, wow, that's amazing. If any reaction has this type of yield, that must be very special. So I actually contacted the postdoc that had done the work and said, okay, this is really fantastic yield. Is this, let's say, your average yield? What do you normally get on, let's say, an average Tuesday morning? He said, well, this in fact was our best yield. Okay. So what is your average yield then? Well, it's roughly 99.999+%. And then I thought, okay, this is a reaction I really want to get into. So the first thing that we did with this type of reaction, we did it together with a variety of groups, including those of Moses and Sharpless and Wu, starting from this SF4 compound, where the sulfur is highly electropositive. This reacts with difunctional, so with diamines, and forms a sulfur-nitrogen double bond, kicking out two of the fluorides. This entity then has two fluorides left. And interesting, what we were able to show is that first, when you react this with a silicon-protected diphenol, this will react, the oxygen couples to the sulfur, kicks out one of the fluorides, and then you get this SF-containing polymer. And there's no trace at this stage of reactivity of the second fluoride. As a result, we can first make this, and then in a subsequent step, react the polymer, basically post-polymerization modification, and basically functionalize the polymer at will. So what was novel here was first that this included a novel suffix linker, and secondly, that it provided a suffix-based polymer that itself could be suffixed again. The reaction basically works for any of these species. So whether you have basically irrespective of what you have between these SF2 moieties or the nature of the diphenol, we could make a variety of these polymers rather easily. And also on scale, you see here a 200 milliliter Erlenmeyer, and it could be basically rapidly filled on a 100 gram scale. So reaction is extremely versatile. Let's focus first on these two, and I'll show you the structure in a bit more detail. This one has a SF2 compound in which the two phenols are bridged by SO2, and the phenol bridged by a carbon with two methyls. And I'll come to the structure of this one a little bit later. So the first compound, we could then functionalize after the polymerization with a big variety of moieties. So for example, you could couple this via a subsequent suffix reaction with a moiety that induces aggregation-induced emission. And indeed, the resulting compound does display that feature. You could also couple it in a suffix reaction with some moiety that contains an alkyne, and then the alkyne can be reacted in a quark reaction, basically adding some nucleoside. There again, adding biological functionality. Another example here is that we added vancomycin and studied the antibacterial properties of the polymer. Or finally, suffix could be used to couple ferrocene containing moiety, and then of course you get a species that are redox active. And again, significant amounts are really easy. So we demonstrated a wide variation of effective follow-up post-polymerization. Reaction basically allows functionalization at the backbone. So you see directly that the sulfur is involved in the reactivity, and the reaction is really mild. So this displays true modularity as is advertised for click reactions. Now, these polymers by themselves also had a very interesting structure that we displayed in detail for the species over here. One of my coworkers, Sidhu Pujari, put this structure, just take the ChemDraw structure, copy, put it into Chem3D and then in Material Studio, and within 30 minutes, he had this structure. That's amazing. Like, it looks like the fusilli drawn over here. And it really has this circular ID. Yeah? So if you would look along this line, basically you would see this. And this only took about 30 minutes. To prove it experimentally, it took us 1.5 years. And for that, we used a combination of AFM, SEM, scanning OJ, and TEM. And I'll show you the outcome. So if you take a polymer solution, and you take an AFM, you see many species that are, let's say, about 4 nanometer. 4 nanometer is much, much bigger than the thickness of the wire. So indeed, that corresponded roughly to this. So that could be in line. But not only that, if you then zoom in into your AFM, you see, indeed, these kind of spirals coming up, as you would expect for fusilli. And this is not an artifact. As one can see, if you take both a scan here outside the polymer, you see actually here an individual polymer molecule, outside the polymer and on top of the polymer. We see that in display number F. Outside, that's the thin orange line. We see basically it's flat. But when we do the same thing on the polymer, we see it's going up, down, up, down, up, down, precisely as what you would expect, up, down, etc. Now, if one looks at such a polymer by SEM, scanning electron microscopy, you see wires like this. Of course, you may think, well, you know, that's clearly, there we look at wires, there we look at polymers. But the question is, how do you know? Oh, why should you be convinced that this is not something else that you see? So what we then did is we took the SEM image, and here you see a SEM image of an individual wire, and then do scanning OJ. And scanning OJ gives you the elemental composition at that location on the surface. The wire is deposited on gold, yeah? And surrounding the wire, we only see gold. But lo and behold, if we then look for the presence of oxygen, we only see it on the wire. Carbon, we only see it on the wire. Sulfur on the wire, nitrogen on the wire, fluorine on the wire, no other elements that we could see on the wire. In other words, you see precisely elements that you would expect for a polymer like this. So basically, this proves both the structure and the composition on a single polymer level. And that we thought was really a step forward. Now, it's interesting to figure out, why is this possible? Why can we first make the polymer and only afterwards do the functionalization? And the first indication why this is the case was shown by DFT calculations. So we take the SF2 compound, then indeed the driving force is mild, yeah, and the barrier is very low. Basically, you put them together, they react. If you now take the product of this and do the second reaction, again, you see the driving force is mild, but not so different. But the barrier is significantly higher, still pretty low, so you can do mild reactions, yeah, but significantly different from this one, that one can first completely finish the first step, in our case, the polymerization, and only then do the functionalization thereof. So this big difference in delta H dagger yields polymers and prevents network formation. And interestingly, so this was a study together with Sharpless, and Sharpless thought, what we really need is silicon functionalization on the phenol, otherwise the reaction shouldn't work. But in fact, if we try to do the calculations with that moiety attached, we did not find any transition state. And so since then, there actually is a big debate in suffix chemistry. What is the role of this silicon protection? In some cases, it definitely seems to help the reaction, but how it works is still something to be discovered. Okay, so that's the red flag. I'll come back to that. Yeah. Since then, also some work has been done to get improved access to this multimodal chemistry. And the reason for that is that the SF4 compound that is needed is actually not commercially available. In fact, it's extremely reactive and extremely corrosive. And so only in very special labs people can make it. And you need to be really provide a lot of control to use it in a safe manner. Okay, but once you have it, when you have access to such material, then of course you can get these species that are highly functionalized. But then, since it's then of interest to look for an alternative, last year both the group of the Borggraaf and de Marel in Leuven and our group in Wageningen came up with alternatives that are much easier. The first group starts with SOSL2, Cl2, and then using a lot of silver fluoride gets access to this SF5 compound, which indeed reacts like this. So nicely providing you access to something similar to the compound that Charlotte says. However, the reaction required lots of silver and provides you with this. That means the two fluorines are identical. We thought, okay, apart from having this dual reactivity, can we also introduce stereospecificity? And lo and behold, that's possible if we start from this NSO compound reacted with xenon fluoride. Then we generate the SF2 compound, but then in the second step reacted with the imidazole. And then we generate this compound. And now we have the sulfur. There's 4 different substituents, and therefore it's chiral. First, we will be able to react it on the sulfur fluorine bond, and then after methylation of this nitrogen, we can actually react the second one as well, providing both access to species that are functionalized in a different manner, both here and there. Actually, given the short time, I won't have the chance to go further deeper into this, but we wrote actually an entire ongoing paper on this last year. But here you also see the first time that the word enantiospecific pops up. So, coming back to the definition, click reactions are reactions are modular, wide in scope, etc. Be stereospecific and use no solvent or water. Okay, the modularity we showed. But what about the stereospecificity? Well, if you look at a typical click reaction, and you may have your favorite one, actually, very often they are either involving a sp2 or an sp hybridized carbonatum. And as a result, there's no intrinsic enantiospecificity. Sometimes, like in the Diels-Alder, you could have enantioselectivity by the use of auxiliary groups, but that is not really what a click reaction would want to go after, because then you have to put it on, the reaction, and remove it again. So our thought was, can we develop stereospecific click reactions? And lo and behold, we could. For that, we take these sulfur fluorine compounds called sulfon imidazole fluorides. There are 4 different substituents, therefore chiral, can be made in a chiral fashion. And whereas the traditional suffix reaction replaces the sulfur fluorine bond by the reaction of the oxygen onto the sulfur with this SO bond, and therefore form this this sulfur atom with 3 oxygen bonds. What we have here is, due to the difference in substituents, we now can couple the oxygen to the fluorine, and if this is if we could make this in a stereospecific fashion, then we could show the reaction actually produces one and only one of the enantiomers of this. So this was actually the very first enantiospecific click reaction. The reaction actually has a very wide scope. So if we take basically any phenol in the presence of some base, room temperature, we get it. So standard phenol reacts within about 5 minutes in high yield, isolated yields between parentheses. But my favorite example is this one. Even if you have an ortho T-butyl, which is a beast in size, then the reaction still gives more than 95% yield. The only thing is that it's a bit slower. So it takes 40 minutes rather than 5 minutes, but still the reaction works very well. In other words, it indeed has a very, very wide scope. Now, what about this enantioselectivity, enantiospecificity. So if you take one of these species, you take a chiral compound, then if we just take the reaction and treat it with a base like DBU, we saw that there was enantioselectivity, but not enantiospecificity. And if we take a poor nucleophile, so if the nucleophile has a nitro moiety here on the ortho position, then the EE of the product even went down to less than 2%. In other words, all the asymmetric character of the reaction was lost. So then the question was, what's going on? And then both us and James Bull in Imperial College independently came up with this idea. Namely, if we not use here the base with DBU, DBU is a base and then a phenol, but first prepare the anion, in this case as a sodium salt. In his case, he used lithium ion. Then what we discovered is then the E of the reaction actually goes up. So no more than 99%. So it really is enantiospecific. And the idea behind this is that the sodium captures the fluoride that's released in the reaction, and that fluoride actually can react onto the starting material, and that would otherwise racemize. So if you capture it rapidly, then we show the EE is in fact over 99%. Therefore, we proved that this was indeed the first intrinsically enantiospecific lick reaction. Very happy with that. So what's going on in this reaction? Well, why is it such a nice reaction? It's a nice reaction because, first of all, the barrier is very low. So we calculate it's roughly about between 2 and 3 kcal. But also the driving force is small. From here to there, it's only about 5 kcal. So we have a reaction that's fast and a reaction that's also still gentle, without producing lots of heat. What these calculations also showed is that if we look at the transition state, we need to understand that the oxygen of the phenolate reacts onto the sulfur, kicks out the fluoride, and the suffix reaction proceeds with inversion of stereochemistry. Then, since this reaction was shown to be enantiospecific, we thought, okay, can we also do this on a polymer? Because if we would be able to do that, we would get polymers with a fully chiral backbone. So no chirality in the side chains, but we could add chirality in the backbone itself. So if we start from a di-SF compound, react it with a diphenolate, get these polymers. In fact, we could show that this works rather gently, wide scale of these red and blue boxes in warm citronitrile, and the reaction just proceeds like a charm. Now, what we then did is we take a few of these di-SF compounds, and each of them could be made in a chiral fashion with high EE, then use with those species the polymerization. And lo and behold, what we do get is, again, high yields of polymers with high molecular masses. So the polymer weight, the Mn is roughly 238 kilodalton, 177, 173. Some are less high because of the lower solubilities, and then this stuff peters out of solution and can't continue with the polymerization. But if that's not the case, we see the EEs, we see the molecular masses pretty high. So the reaction works very well. But then the question is, of course, well, but is it enantiospecific? Well, very often in the literature, what you see people do is you take the starting material, hit a monomer, react it, we get a polymer, and then we take a CD spectra. So circular dichroism of the monomer, we see that if we start with the S monomer with this CD signal, lo and behold, in the product, we get the inverse. And if we take the other, we get also the inverse of that one. In other words, we get inversion. Now, that clearly shows inversion of configuration of polymerization, but it doesn't show that it's enantiospecific. Merely, this shows that reaction is enantioselective. So how can we prove that reaction indeed is enantiospecific? So for that, we synthesize this polymer that has this moiety in there. The polymer itself, again, peters out of solution, so the molecular mass is not so high. But the key thing is that once you have this, then in the subsequent post-polymerization treatment, we could cleave this SS bond, methylate it, and then we get this product. And then we could just by chiral HPLC analyze to see how many enantiomers there are. And lo and behold, there's only one. So we here have shown that the polymerization itself also proceeds with over 99% enantiomeric access. So we have a chiral suffix polymerization that is indeed enantio specific. Very happy with that as well. Now, the next step then was, well, can we somehow invert its stereochemistry? Because going back to this, if we would start with one, configuration, you would get inversion. That means if you would have, if you would want the other configuration, then you would have to synthesize the other enantiomer of the starting material. And that's a bit cumbersome. So we thought, can we avoid that? And for that, we developed another sulfur exchange chemistry, namely the sulfur phenolate exchange. We figured that if this reaction is so smooth, in which a nucleophile can kick out a fluoride out of sulfur, suppose we would take a phenolate, which itself has a very strong electron withdrawing group. Then this should also be a reasonably good leaving group. And lo and behold, if we add a phenolate onto this, indeed, we get one and only one product, and it's pretty fast as well. We could show this again for a wide range of phenols. Again, you just add it, room temperature, you stir, you're done. But now the trick comes. Of course, it means that once we start with one enantiomer and in suffix get the sulfur with here the oxygen pointing forward, if we first do the click reaction with the nitrophenolate and get this intermediary product, and then subsequently do the other, the phenolate reaction. So now we do a sulfur phenolate exchange. This is a suffix and this is a sulfur phenolate exchange. Then we get the sulfur with the inverse configuration. So what it shows is, first of all, we have continuously high yields also for this click reaction. In the second step, you can avoid fluoride, which is nice for some applications, but this is very interesting, namely that we can invert the stereochemistry while just starting from one compound. And again, we were able to show that this works for a very wide range of materials. Since these reactions are controllable in terms of their chirality, and since they are so effective, this brought us to the idea, can we use this to make chiral sequence-defined oligomers? And from there, also sequence-controlled polymers. I'll start to give you an idea about what we're doing by showing you this oligomer, which has the sharpless configuration. I'll come to the reason for this. Here we start with this species and we basically grew from right to left, where every time we click one item on. Yeah, so every S is a chiral sulfur. We can add, when we take this phenolate as a start, we can add this moiety. Then from here, we do another click reaction, etc. The nice thing about this is that we can control the stereochemistry per site. And it was nice to take this as an example because we generated it with the stereochemistries S, R, SS. And of course, you can easily remember that as sharpless. Now, you can also do this on a longer scale, and we could generate oligomers up to 8.5 kilodaltons, so quite substantial. And the yield of the Suffinex reaction, especially the Suffinex reaction, doesn't go down so much. So it continues to be pretty high. In fact, Suffinex works better than Suffix in here. This was the basis of a recent paper. Now, why was this interesting? The first time I showed this to a wider audience was actually in 2023 in a meeting together with Sharpless and with Jia Dong, who was the host in Shanghai then. And we really had a good laugh about this, the sharpless configuration of the oligomer, the same one as I just showed. Now, to go a bit more in detail, what is going on here? When we start with a chiral SF2 compound, so we have here SF, and here we have an SF moiety. We react one of the sites in an asymmetric suffix reaction with a phenol that is then going to be used as a cap. And next, we can do an asymmetric suffix in couple. we get now this moiety. This can be reacted again and again. And if we just vary the building blocks, we get these sequence-defined oligomers. We could do this, and then basically the PhD student thought, should we continue any further? And said, oh no, we've proven the point. Up to 12 reactions in a row, each of them functioning with functioning well with high yields, and where possible, shown initial specificity, we're finished. So, taking such sequence-defined oligomers, what we then did is, of course, if you would want to make a polymer out of this, you would have to be able to react both sides. But here we had a cap. By clever choice of a sulfur-based cap, a simple activation by oxidation turned this dormant cap into a reactive cap. And then this species, if you then react that under Sufx or Suffinex conditions, indeed proceeds with polymerization. So this is a very nice way to demonstrate. Here is just shown for a species with 2 blocks, but we've done it also with 4, and you could easily extend it to 6 or 8. So you really get sequence-controlled polymers that are tunable in their, not just in the nature of the building blocks, but also with regard to the stereo centers that are present in there. This we started out with this paper over here. Very recently, we extended this to a paper in the new flagship journal Chemistry Europe. Okay, final goal: no solvent. Why would you have no solvent? Well, first of all, very often if you do a reaction, we say, well, we have very high yield, etc. But over 95% of our reaction mixture is going to be used as solvent, which we then afterwards have to get rid of. Remove, rotate away or so, but still it's gone. And that mass is then not used effectively. So a challenge is then, can we do the same type of reactions? Can we develop reaction conditions in such a fashion that we don't need a solvent? And here mechanochemistry comes in. So what we're able to show is if we start with chiral SF and also SCl species, we're able to do enantiospecific reactions both with oxygen nucleophiles and with nitrogen nucleophiles. We do this by just simple bases, sodium hydroxide and a little bit of cesium, for detailed reasons I won't get into. But then we get very high yields of products. And what do you then do in practice? Well, you just put the reactions together and literally you shake in a ball mill. We are able to do this with both Sufx reactions with sulfur, chlorine exchange with Suffinex. We don't need solvent. The reaction is enantiospecific. It's very high yield and has an extremely low E factor. If you have any interest to try any of your own reactions of interest in this fashion, feel free to contact me by email. I won't have the time to go into this in much detail, but we just published a paper in Chemistry Europe on this. This is really something that I think is a big step forward to 21st century chemistry. So this brings you to conclusions. I hope I've been able to show you that sulfur-6 exchange chemistry allows for multimodal Sufx, that it provided the first intrinsically enantiospecific leg reaction, and then also allowed for click polymerization with chiral backbones, that it provides an entry into solvent-free click chemistry and also into sequence-defined chiral oligomers and polymers. With that, I'd like to thank you for your attention. But before we wrap up, I'd like to give a big thanks to many of my contributors. I specifically want to mention Fedor Miloserdov, Sidhu Pujari, and Divoje Sevkirk in my team, the international collaborators on the multimodal Sufx chemistry, and the Euros and RMBs came from a variety of sources as indicated over here. With that, thank you very much, and of course, I look forward to any questions and comments.
Thanks very much, Han, for a very spectacular lecture about the Sufx, Suffinex, and chiral polymer reactions. Okay, questions or comments via the chat box. And I see here the one who says, thank you, Professor Han, for the nice presentation. The first reactions involve fluorine elimination. How do you manage the large amount of fluorine produced? So the fluoride that's produced, how is that removed?
Okay, it's a good question. Basically, what is key here is that you start out with, in our case, that we start out with a sodium phenolate. And that means that we not only generate a more reactive phenol, because we already have it deprotonated, but also we have a sodium. And in the solvents of choice, that means that these sodium atoms, sodium ions, couple very effectively with the fluoride, therefore get out of solution. And therefore the fluoride is not involved in any racemization anymore. So capture of fluoride is guaranteed in this fashion.
And I think that's also, could it also drive the reaction forward, or do you say no, the reaction runs anyway?
No, the reaction runs anyway, because if you do it with DBU, the reaction also
true.
works.
So
It also works smoothly at room temperature at high rates, but then you lose the initial specificity of the reaction.
Yeah. In terms of enantiomer selectivity, is it difficult to start with it's difficult to make the enantiomer pure starting monomers? But maybe I missed that.
Let's say it's getting easier. And that's not by our efforts. Yeah. So basically, after 2020, when we published this first enantiomer specific click reaction, then I think many synthetic groups started to realize, oh, there's something here. And by now there are 3 groups, two Chinese and American, that have independently developed methods to make this in a enantiomer specific manner. Yeah? No, enantiomer selective manner. We get compounds with high EEs.
Okay.
So it's getting easier.
Yeah.
But okay.
Okay. More questions or comments? Now, for the people who attended the whole webinar, my compliments, because this was quite lengthy, but I enjoyed it tremendously. Thank you, Han, for your contribution. Very nice.
It's a pleasure. Thank you for organizing it, Adrie.
Okay. Thank you. And
I think all these applauses are for you. Yeah.
Oh, yeah. Nee, then I'm taking those. Okay. Yeah. Wow. Thank you, because I thank all the speakers for their excellent lectures, and I sincerely also thank Jeffrey van Vloten of our University Audiovisual Services, because this webinar went very smoothly. And I hope to see you back in December with a new edition of the Organic Chemistry Highlights, and then from another European country. Okay, thanks for joining. Bye.
Okay, bye-bye.